Hello, everyone. Welcome back to Ask a Scientist Gaming, where we combine mediocre gameplay with expert science. I'm Ken Hansen, professor in the Department of Chemistry and Biochemistry at Florida State University. I'm a photochemist slash photophysicist. I shine light on molecules and materials and try to track what happens to that light energy and hopefully do something useful with that light energy. But more importantly, joining me today is Dr. Jim Fadool. Jim, if you wouldn't mind introducing yourself.
I'm a professor of biology, and I study the biology side of light. So I study how vision occurs and the cells that are involved in converting those photons into the chemical and electrical signals of your brain. So why you see colors, why you see black and white, why you don't see well when you walk into a movie theater, or you don't see well when you walk right out into the sunshine.
And what do you typically teach?
I teach genetics or developmental biology, and I teach what we call baby bio. And so that's biology for non-majors. And we teach it in a 1,000-seat auditorium where we hold operas and concerts. And where I saw Willie Nelson play, which was a phenomenal concert. That was about 20 years ago.
You can say you shared the stage with Willie Nelson.
Well, I don't know if I really shared the stage with. I shared the stage with Greg Erickson, the dinosaur guy. He's almost as famous as
He's
Willie.
almost as famous.
At least in some parts.
I like it. All right, equally important, what game are we starting with?
Why don't we start with, I cannot pronounce the names properly, Galaga.
Galaga, all-time classic. Let's do it.
Well, I'm a classic. So.
It's one of those, arcade games that never gets old, actually.
You know, you move right or left and you shoot.
Yeah, but it's way harder than most modern games. Yeah. The irony of it. It's got 3 buttons and yet.
But there's no secrets.
Yeah.
Secret chambers, you don't have to remember when to turn right, left.
Yeah, there's no easy level.
Got pinned in the corner so soon.
So we're hanging out last week before classes start. Got your Canvas page set up already?
I have my Canvas page set up, posted my syllabus, an official syllabus.
Oh, one of the university approved.
We're en route to doing that, but it will probably become the standard. Yeah, syllabi nowadays are much different than when I was in school or when I started this biz.
So it's much more of a legal contract.
Yeah,
right.
Yeah, they take this seriously now.
Yeah. Which I respect. I mean, students should know what's going on. The thing that gets me is when you have, 10 different sections of the same class.
Now you'll have to start it.
There we go. 10 different sections of the same class, but to have different course curricula and course content. And
yeah, sometimes that actually is a good idea. So when you're writing or you're taking a lit class, it's nice to have a choice of authors or types of literature.
Well, that's a good point.
But then in other classes, you want your Chem 1 to be Chem 1.
Mm-hmm.
Yeah, then I bought it.
That was good, though.
That was luck.
Take that luck. All right, Jim. So it's been almost 4 years since you've been on this.
Yes.
Game, which is crazy. Time flies. It's you spent at least one of those years on sabbatical, which we'll get back to. But we'll ask you the same question we asked you 4 years ago, but for a new audience, or potentially at least a couple of returners. But more for the sake of YouTube. Like, most students think that we were destined for this job, and we knew from the start we were going to be professors working on our various research areas. When in reality, we had a lot of twisting and turns and forks in the road and decisions that put us one way in life or another. So the question for you, starting with, 6-year-old Jim, where he is today, what were those forks in the road? We're looking back in retrospect, what distinct moments made you who you are today?
Oh my gosh. Okay, 6-year-old Jim.
Yep.
Okay, maybe 9-year-old Jim set up a lab in the basement of the house. It was a dark room. And it wasn't a fancy house or anything like that. It's just that the prior owner put a dark room in it.
Sometimes you need a dungeon. I get it.
Yeah, that's right. And it was snowy out half the year, and I liked to read encyclopedias, and I was not good at sports.
Yeah.
So what are you going to do? So I set up a lab. But that, it took me about 20 more years to figure out this is maybe a career you could have. Because who knew a college professor when they were a kid?
Yeah, exactly.
And I knew one person who went to graduate school, and she lived across the street, is an old, neighbor that maybe 10, 12 years older than I. And she quit because she said it was horrible.
Oh.
And so she got a master's degree in microbiology, and I was maybe 12 at this time. And, so I don't know how I ended up here. What else are you going to do? I had a biology degree.
So you graduated high school. You knew science was something you wanted to do.
I like science. I like biology. I took chemistry classes because you have to as a bio major. And I had phenomenal organic chemistry professors. And I decided, well, if I went into science, then I didn't want to study chemistry, though, because I didn't like the stories they were telling at the time. I didn't like the research that they were doing, the little bit that I knew of research.
Yeah.
And I still like biology better. So I kind of merged the two and studied a lot about chemistry. But becoming a college professor, I mean, there's this intervening period between graduate school and faculty called a postdoc or postdoctoral fellow. And I didn't know what that was when I went to graduate school. And there was one on my floor. His name was Ned, and he was in the lab next door. And Ned was very nice. He always asked me what was going on. And finally I asked Ned, and it's like, hey, Ned, what is your job? And he's like, I'm a postdoc. It's like, what's a postdoc?
What would you say you do here, Ned?
Yeah, what do you do here, Ned? Besides, talk to the graduate students and be nice to us. And kind of mentor us a little bit. And so it's like, okay, so then you have to get a PhD first. Then you go off and you a postdoc, and you do more work for somebody. And then you hope to be one of those lucky few that got lucky during... Oh, no, don't want to get caught up there. I have no more people left. And so Ned was a big help in graduate school, and then had to do a postdoc. And the next thing people ask is, well, why do you study vision? And it was completely chance. I worked in a microscopy lab as a graduate student, and my significant other is also a scientist, and she wanted to do her PhD with a certain person at a certain lab. And I had to find a job at that lab. And the only person that would hire me worked in vision. And so
you got to select a name there. That's what it was doing.
Oh.
Now we're good.
So that's how I got into vision. So, it wasn't like I grew up as a kid saying, I really liked light. Though I really did. I thought rainbows were really cool, the splitting of light into a rainbow.
Yeah.
Magnifying glasses, prisms. That was all really interesting stuff to me. And so, my gosh, I have a job where I can, look at a microscope, take pictures of really small things. So it's actually not that bad. Get to play games
this
online.
Yeah.
So I don't think every school has this. Ken could go on tour.
Yeah.
Ask a Scientist Gaming, Utah.
What's kind of fun is we've had visiting speakers that I've invited to be on. It's kind of fun. Yeah.
That would be cool.
Like the night before seminar, let's get drunk and play video games. Oh, it gives you percents. That's kind of cool.
Yeah, it does. It makes you feel bad.
No, it lets you
actually
track your progress.
Hey, and if somebody's out there and you think this could be your job, I was a B student at best.
Oh man.
Right?
My lowest grade in high school is chemistry.
Yeah, I'm a developmental biologist. I study development of the visual system, and I never took a class in that field.
Yeah.
So, you just kind of get to make stuff up as you go along, which is kind of cool, too.
I don't know your opinion on this, but I think the dirty secret is most of your learning does not come from a classroom.
No. It can be really efficient learning in a classroom.
Yeah.
Right. So that's, supposedly you have an expert in front of you telling you what are the most important things you're supposed to know.
Which you don't appreciate until you're an expert yourself.
Yeah,
I wish I would have
paid more attention to the expert in the field.
Like, I should take your biology class.
And you said, well, I could probably I'm due for a chemistry class. That whole pH thing.
I should invite you to my pH section. So I'm Gen Chem 2 this semester. So
oh, really?
Yeah. I can tell you exact days. Contra, welcome back. Thank you for joining us. Contra says, good evening, docs. The docs.
Welcome back.
Yeah, so Jim, I don't know. Contra, I don't know if you were around. Oh, wow, the tier 1 subscription, subscribing for 14 months. Thank you, Contra. We really appreciate you. Help us with our drinking habits. So cheers, Contra.
Cheers.
You let us play video games.
I finally got 2, shooters, and I don't want to blow it.
Oh, yeah, no worries. Priorities.
Wow.
Because you can get up to 3? on this.
Mm-hmm.
But cheers, Contra. Thank you as always for joining us. If you have questions for Jim, we're going to talk about neurobiology and zebrafish genetics, human genetics.
Yeah, why not?
Talk about, well, basically anything you want, but
Food.
primary expertise is in biology and neurobiology, and particularly related to vision. So yeah, if you have questions in those areas, he'll definitely answer them. Or food or video games, we're happy
Let's see, what other jobs did I have? I drove a big yellow school bus.
Oh, wow. When was that?
When I didn't know if I was really going to get into graduate school, and I had a job teaching science at a camp. They needed a bus driver. And from a previous job, I had a chauffeur's license. I just had to get my bus driving certificate. And they paid for it. So, why not?
That's awesome. Man, that is a question I should ask guests. What other jobs have you had? Because I think that's underappreciated too. Like some students don't know we had a life before this, but sometimes that life is winding back. My favorite was for 2 summers I did help build rigging for playhouses. So the pulley system inside the theater, I helped build those. So there's a lot of universities and high schools and stuff like that.
Yeah, one of the I cooked as a chef in Rhode Island on A1A, so it was a resort area. That was a lot of fun. Restaurant workers don't get enough respect.
Oh my god, what a terrible job. Sorry.
And they, but the great camaraderie.
Yeah.
But it's challenging, right? Everybody else is out having a good time on holidays and weekends, and you're working, especially if you're good.
Yeah. Now, there's always a Reddit thread like, which job seems glamorous but really isn't, and chef is always up there.
Yeah.
Because movies make it look sexy most of the time.
Yeah, yeah.
Like you're creating new dishes, not grinding the
No, it's we used to count how many burns and cuts do you have on your arms. It's fun though. There's a great crew to work with, and I think it's also underappreciated that that's a path many take because like the chef I trained under, his mom passed away when he was young, and his dad only made meatloaf. So he figured, I want to eat more of the meatloaf, so he got a job at a dishwasher where they fed him things other than meatloaf.
There you go up and down.
I see that, yeah.
I mean, doesn't really matter, but...
Let's see, what am I? We'll just use Jim.
Awesome. Immortalized until we restart the game.
That's right.
It's like unplugging the arcade. Isn't there a Seinfeld episode where he's trying to save his Pac-Man score or something?
Yeah.
To a generator. I don't remember what game it was. If anyone knows, do you remember off the top of your head?
I don't remember.
It was one of the classic arcade games, but total respect for that. All right, Contra opening strong. I have quite a few questions on this one. My partner for 7 years has retinitis pigmentosa.
Yes.
And getting into the SNPs, we found that her mutation was in the TULP TULIP1.
TULIP1, yes.
Tubby-like protein
Tub.
gene. Yeah. My first question is whether Dr. Fadool does any work involving retinitis pigmentosa.
Wow. So retinitis pigmentosa, for everybody, it's a disorder that individuals typically lose their night vision first. And so you lose a cell type that we use only in starlight rods. Normally we use cones because they are good in color and they're really fast. And so while playing video games, it's all cone driven. And when the rods die, then the cones start dying. And there's maybe 100-some different genes that, when mutated, cause this form of retinal disease. And TULIP or Tubby1 is one of those. What we've done in my lab, I work on a model organism called zebrafish, and we've made fish models by mutating the fish gene, not Tub1 or Tub-like one, but another gene called rhodopsin, and that's the most commonly mutated gene that leads to retinitis pigmentosa. So we've made these fish models, and we've mutated the gene to look like the human gene using CRISPRs. And we put in mutations that match those that are in some human families. And the goal is that if there's people out there that do drug screens and chemical screens, then they could use these as models to try to look for ways to either protect those cells from dying or decrease the secondary changes that take place.
Interesting.
So yeah, we actually have done that, and from the very generous support by a foundation called Foundation Fighting Blindness. Is it okay to give Foundation Fighting Blindness a plug?
Shout out. This stream brought to you by.
No, they were very generous, and it's a lot of their funding comes from families, and then if people have patents that generate revenue either through drug screens or other mechanisms, then that portion of that always goes back to the Foundation Fighting Blindness. So no, that was a great question, and onset is different depending on the gene, depending on the family, and depending on the person. So there was a family that a friend worked with. He's an ophthalmologist, and most of the people in the family started showing signs when they were young adults. But one sister, she retained her vision into same mutation into her 60s, and she was actually a commercial truck driver.
Wow.
And then she stopped commercial truck driving when she could no longer felt it was safe.
Yeah.
But she had the same mutation as her siblings. And so this variation, even in families, affects how it affects individuals.
And so it's not predictable. There's just a probability
everything's a probability. There's no absolutes in many genetic diseases. There's a few that are, the probability is quite high.
I see. Man, that's crazy. How soon does that typically onset?
RP, it can be juvenile. For most, it's adolescence.
Wow. And they just like low light vision. They just
low light vision goes first, but once we lose those low light vision receptors, then we start losing the cones, which are fewer in number, so they kind of have a community effect. And there may be, what would this be? AAA. That was a
already
top score. Yeah, Jim's on top. We don't want to displace Jim.
Yeah. So in that question, he asked, getting into the S&Ps.
S&Ps.
But the details of it, I was wondering if that was a genetic test or something.
Maybe not. Is he
getting into
listening?
The S&Ps? We found that her mutation was at the TULIP 1.
Yeah.
Like protein. So S&Ps might be just slang in that case.
Yeah.
S&P, single nucleotide polymorphism.
Oh, yeah. SNPs. SNPs. I thought. Yeah.
SNPs is the...
SNPs, we say single nucleotide polymorphism. Oh, I wasn't reading the screen.
Yeah, no.
If I would have looked at it. No, that's...
You got to focus there.
Yeah. No, so SNPs.
SNPs, that's the... Okay, I apologize. I don't know the nomenclature.
That's, so SNPs are... the human genome is billions of bases, and we carry... As individuals and as populations, there's variations in the genome, and we call these when they're just single bases, right? So it's all these letters, A, T, G, C. And when there's single variants in some individuals or families, then we call that a SNP, because most of people in the population will have one flavor, but then there's others that will have another letter or another base at that position. And we can use those for mapping. We can use those for doing family lineage. We use those for population studies, and those can be used to track disease alleles. So getting into the SNPs, we found that the mutation was in, so using SNPs, they would have narrowed it down to the gene, because they can look for specific changes, these single nucleotide polymorphisms, in the genes that are known to cause retinitis pigmentosa. And so since many of these genes have already been identified, then they can look for these variations or polymorphisms.
All right, contrast follow-up. What you described was right in her case. TULIP, is it shorthand TULIP?
TULP1
TULP is like 1.5% of cases, so it's probably not getting worked on first. How does CRISPR actually work?
How does CRISPR actually work? So CRISPR is really cool, and originally was derived or is found in bacteria. So we never thought bacteria had a great host defense system against infections, but it turns out they have this CRISPR scheme where if a bacterium is infected with, say, a bacterial virus, then a little bit of that virus can be plugged into the genome, a little bit of the virus genome, and then future generations of that virus, when they're infected with that bacteria, with that virus, sorry, got that backwards. Future generations of the bacterium, when they're infected with that same virus, then they can make a tiny little bit that looks like the virus, and it will bind to it, and another protein called Cas9 will cut the virus genome. So it effectively renders it ineffective. And then several different groups figured out how to harness this for manipulating other organisms. So we can make a copy of any gene and hook it up with this Cas9 and then put it into a cell, and the Cas9 will cut that gene that we're targeting, and then we can either let it repair on its own, or we can add lots of copies to make the change that we want. So CRISPRs are, I don't know if that answer helps, but it came from bacteria, a way that they protected themselves from infection, and then we can harness it to actually make changes in genes that we would like to make. And some are random changes in the gene, and others we can be directed. And that's what's now used, or it's starting to be the platform that looks promising for certain types of gene therapy.
I was going to say, there's at least some FDA-approved use of this.
There is, and how it works is in those, though, they try to deliver something much more precise and short-lived. And so figuring out particles rather than using viruses to move the CRISPR into a cell, because then it's a virus that has to infect the cell, then there's ways of trying to couple it with other things that are nanoparticles, really small particles of grease droplets, and then those will get it into the cell, but it doesn't require using a virus to transduce a cell. And so that's for retinitis pigmentosa. I don't think any have made it to the clinic yet. There may be several early clinical trials, but that's an area where there are a couple viral therapies used for a few diseases. But the viruses do have limits. And so some of these new modes for delivering the gene targeting seem to be really promising, because they wouldn't be limited to only a single use. We could try an individual, if one type of approach doesn't work, then they could use a different type of approach on the same individual, and you can't do that with viruses very effectively. So the limits, not the CRISPRs really, the limits are really the delivery mechanism. But that's common in a lot of pharmaceuticals. It's delivery. How do you package the drug so that it's most effective and the least number of byproducts or side effects?
Opinol, your Dev Bio class was the best class I've ever taken.
Oh my gosh, awesome. Thank you.
That's awesome. We got alumni in the chat.
Wow.
Or maybe still a student. I don't know.
That's the class that I grade the easiest, because if people are willing to sit through it, then you deserve a better grade.
That's developmental biology?
Developmental biology.
It's like third year?
It's a 4,000 level, so I usually get seniors in about a third of the class took my genetics class.
Oh, nice.
And it's the third that liked my genetics class.
They're back for Dev Bio.
And then they come back for Dev Bio because they got it the first time.
Oh, man.
So that's really cool.
Yeah. My question for you, Opinol, is how did you find out about the stream? It was because we tweeted it, right? It's been 4 years since you've been on. We didn't advertise much this time. Well, yeah, welcome back. If you're not following, click the follow button. You'll get notifications about future streams. But yeah, Contra says that's really fascinating. Thank you. All right, Sporty Stats says, my son has albinism, and as we were going to vision camps, it seemed like there were a significant number of children with genetic eye conditions that were from twins. Has there ever been any studies to investigate the correlation between genetic eye conditions such as albinism, LCA, JMD, and twins? Let's start with, what are LCA? Do you know these conditions? LCA?
Yeah, LCA is Leber's congenital amaurosis. So that's again, it affects the light sensing cells, but it's actually in the cells behind the photoreceptors called the pigmented epithelium. they're pigmented black, so they absorb all the stray photons, but they also provide lots of nutrients and metabolism type things for the photoreceptors. JMD, I might need help on that one. I should know my genetic diseases better, especially of the
juvenile
macular degeneration. And I was thinking it was a ciliopathy gene, but it's not. Yeah, so juvenile macular degeneration. So we typically think of that as AMD, age-related macular degeneration. And many of our parents or grandparents have that, and some of those are genetic and some are not. The juvenile tends to be genetic, and so the macula, what we use for most of our vision is this tiny little area called a fovea, and surrounding that is the macula, and that's what we use other than the really close high acuity vision. And it's, again, lots of cones, these color sensitive cells in the macula as well. And age-related macular degeneration, different risk factors, some being genetic, juvenile macular degeneration, most of the risk falls into genetics. But twins are no more likely, in my understanding, than individual children or single births to have an eye disease. There may be some forms that, due to environmental constraints during pregnancy, etc., then it may push twins into that higher risk group, because twins do have higher risk for some childhood diseases.
Interesting.
But that may just have been one of those instances where you see a higher number than you would expect, but then the next week there might be a fewer number. Right. So
anecdotal evidence does not cut it then.
Yeah, but that is interesting, because maybe there is a component, because twins do have a higher risk of some types of disease, or actually expressing those diseases. But I haven't read any literature about it being for these retinal diseases.
Any idea the mechanism behind that? Why twins would be higher prevalence?
Oh my gosh, I think we would need our former colleague from psychology, Sarah.
Yeah.
Yeah,
Now in Canada.
Who's now in Canada, yes. But Sarah actually studied twins. She did twin studies, mostly in learning, but she actually was a phenomenal human geneticist of her own right. And so she would be if I were at work, then I would send Sarah an email saying, "Hey, Sarah."
Yeah, what is the twin studies?
That's right. That's right.
That's pretty cool. And albinism in this case is just the inability to generate chromophores.
It's usually the melanin pigment that's in our skin, our hair, the back of the eye. And so these children tend to have ocular issues for a number of reasons, or even other animals that you see that have the light pink eye. And there's several different reasons for that. Sometimes it's pathfinding from the eye to the rest of the brain, but the photoreceptor cells are at great risk in individuals with albinism, because the photons that are extra don't get absorbed.
And so in that case, they're colorblind, but they still...
No, they're not colorblind per se, because this is a pigment, not a visual pigment, so it's not the pigment we use for catching photons to see. It's the pigment that we use to catch the stray photons that weren't used in vision.
I see.
So it's the black screen, behind the rest of the eye.
That's it. Okay. But it still expresses the normal receptors then.
But they still have the normal receptors. But because of there's lower visual acuity, so proper connections when we're infants, connections from the eye to the brain get refined by having better vision in the eye. So it's kind of a experience-dependent neurodevelopment.
Interesting.
Yeah, and so in children with albinism, then that's one of the challenges they face, is this experience dependency.
Yeah, I was going to say it makes it very hard to study that, right? Because there's too many variables to account for.
Typically, most of these children will wear glasses, and so there's early intervention, as with many of these, makes a world of difference.
Yeah. Sporty stats. Thank you. I was curious because my personal one data point observation, which is not scientific evidence. No, it is evidence, but it's
Actually,
not necessarily a conclusion, right?
But all of science is based upon that one data point.
Yeah, that was
where
weird.
we say that's interesting. Yeah, that's weird. Yeah. It depends on what hour it is after playing games.
Yeah, exactly. And how many Guinness we're in.
That's right. So it's either, oh my gosh, that seems amazing, I better study that, or that was weird. Right?
Sometimes you look the other way at weird things. 'Cause you're like, I want to figure out why that happened.
I'm
tired.
I have enough work to do.
Exactly.
I'll send an email to Sarah.
Oh, man. The number of times that has happened with, like, not necessarily science, but, like, administrative or education-related things. Like, I would really love to know, like, what dictates departmental ranking and stuff like that. Like, you could... and then you're like, no, I don't want to go down that rabbit hole. Opinel has a follow-up. I followed you since my human physiology professor came on the stream about a year and a half ago. I had to join when I saw Dr. Fadool was on. That's awesome. Yeah, that's why you put your notifications on, because sometimes it'll be someone you're excited about. I'm curious who the human physiology professor was. Who did I have on a year and a half ago?
Roberto Vincent?
Oh, yeah, we had... Yeah, he was on.
Does he teach? I think Roberto teaches.
Yeah, he was on 1 year ago.
That could have been it.
Yeah, we've had several Oh, yeah, sorry.
It's okay. Gosh, I have to start my own game this time.
So many quarters.
I did bring my roll of quarters.
It was Roberto. That's awesome. Well, welcome back. Thank you for joining us. If you have questions for Jim, put those in chat. We are happy to catch up on those.
Well, that was an easy guess because there's only so many faculty that teach human physiology, and one of them I know well, and she would never find herself gaming.
Limited the options. I suppose you're the one. You're the one that chooses or lines courses with interests, we'll say.
Well, I can. Chairs have all power.
Yeah.
Chairs of departments, and I just try to make the chair's life a little easier, and by putting people where they want to be. And ideally for a faculty member in the sciences, we don't change our teaching too often. It's a lot of work to put together a new lecture.
Yeah, it really is. It's the new good lecture.
Well, yeah. Well, a whole semester's worth of good lectures.
Yeah.
When you're junior, I remember, I had little kids, so I'd get up at 3 a.m. because after a week, okay, this isn't working.
Mm-hmm.
I have to, adjust. So,
man, especially in that auditorium, the 1,000-person seating, like, you can feel the audience. There's enough people there, if they're engaged or not.
When they asked if I wanted to, I was brand new, and they said, well, go watch a couple lectures. And I watched one person, and it was a fine lecture. And I'm thinking, what the heck am I going to teach 1,000 people at a time? And, we always have to give a dose of, disease-based things. Mm-hmm. And so I started doing kind of a human genetics-y section there. But I watched one professor, and he was just outstanding. His name was Ted Williams. And the last of the 400 hitters. And I don't know if anybody of the audience will get that.
I'm over 40.
Yeah, so Ted would actually dress in costume. And so I don't dress in costume, but,
Wait, what costume?
Oh, he would come in to teach, because we're teaching about how to do science, but we use our own fields. And Ted was a biophysicist. And he was director of the IMB at one time. And Ted studied the packing of molecules in membranes. He came in as Professor Clouseau from the Pink Panther. He would come in dressed in garb like a monk to pretend he's Mendel. And,
Wow.
So he went all out.
That's a commitment.
Oh, yeah. Ted did nothing halfway.
Did he do the accents?
I was just amazed this guy was in costume.
Yeah. Wow.
Yeah, so that I do not rent costumes.
Yeah.
That's more commitment than I have.
See, the key is you need to own them, so you can use it every time you teach.
That's true. That's true.
A collection of costumes. That is amazing.
Yeah, but and Greg Erickson also. He's our he's our paleobiologist in the department. And
Yeah.
he teaches a dinosaur section.
He brings in the femur, right?
He brings in the giant femur. He has a cast of a tyrannosaur femur that he brings in, just so people appreciate how big these things really were.
Oh my God. It is hard to grasp that.
Yeah. Yeah. I'm just amazed that we find as many fossils as are found. Imagine all the parts that are they decayed normally, right? So the odds of making it to be a fossil has to be incredibly low.
Yeah. Oh, that's fun. All right, Contra has a follow-up. What role might epigenetics play in all these things?
In the diseases?
Yep.
Oh, that's a great question too. So epigenetics is not traditional genetics in the way that, one gene encodes one protein that has a specific function. Epigenetics is really how the genome, one way it marks genes to be turned on or off. So that's a complex story of turning genes on and off, and epigenetics is one of those ways that genes are turned on and off. And epigenetics, it kind of looks like this magic way that the environment impacts not just ourselves, but future generations. So, we know that individuals who smoke, then they have changes to their genome in these epigenetic marks that people that never smoked rarely have. The twin studies of the two astronauts, and one's in Congress.
Yeah.
And they were astronauts, and one stayed on Earth the whole time, and the other was in space for many, many months. And then they did studies on the changes in lymphocyte epigenetics. And the one in space and the identical twin on Earth, the one in space had different changes as they aged than the one that stayed on Earth.
Man.
So that's, and there were fewer differences when they were both living on Earth. But even identical twins have different epigenetic marks because their life experience, even in utero, is slightly different. And some of this is just random. It's just kind of that chance that we play, rolling the dice with biochemistry of cells. I'm not a quantum physicist, but the quantum folks tell me, well, it's all quantum mechanics.
50-50 shot. It either happens or it doesn't.
Yes.
That's how statistics work, right? Sporty stats.
That's right. And quantum mechanics is, that's all I know about is that, okay, it may or may not happen.
Yeah.
And if it does happen, then it happened.
Yeah.
And
quantum information science is either it happens or it doesn't, or it's somewhere in between.
Yeah.
I like it.
So the epigenetic control. So that's like that family with retinitis pigmentosa. The woman who escaped it until she was much older than her siblings. That may be epigenetics, or it could be regular genetics, and she just had a different mix of genes from her mom and dad than the other kids. So it suppressed it, and we don't know.
And so how do you study epigenetics then?
Yeah, so you have to get Jonathan Dennis on here. So
he's on my list.
He's on your list. Yeah. And so how do you study it? Well, it's biochemistry. And so these marks can be tracked with biochemistry because the DNA, even though the bases, these A, T, G, Cs are in the same row, they can be modified. They can carry like an extra OH or alcohol group. They can carry an extra carbon group. And these are marks that are added either normally, so that genes are turned on or off, or they can be added by environmental changes like aging, smoking, and other stressors. And we can track that just using simple biochemistry. But to do it for a whole genome, then you have lots of data. So then you have to do computational work to keep track of your data.
Oh, that's fair. All right, Contra said, I asked the question about the DNA methylation, thinking about how if my former partner had an extremely high stress event, she would suddenly lose a chunk of her vision.
Wow. So physically, people with RP are more prone to retinal detachment. So if it's a physical stressor, things like that, or things that raise blood pressure, etc., then that may happen. And my understanding, I'm not an ophthalmologist. There's specialists that work just in RP that are ophthalmologists. But long term, we do know that some historical events or lifestyle changes can impact epigenetics. And but when we have a causative gene, then it shifts that probability somewhat, and it varies from gene to gene. Does that make sense?
Yeah. Makes sense to me. You can put an additional question in chat if you had it. Old man Felonius said, Galaga, great game to fire particular neurons.
Those neurons haven't fired for 4 years.
And we are back with Jim Fadool playing video games, talking science. So what a great time to join us, Old Man. Thank you for joining us. Old Man is actually an education-related streamer. You should check him out. There's a link right there in chat. I think, if I remember correctly, physics? I want to say physics, but it's been a while, so you'll have to correct me if I'm wrong on that one. But thank you all for joining us for Ask a Scientist Gaming. Mediocre gameplay, expert science. Guest today is Dr. Jim Fadool, who's an expert in neurobiology, in particular zebrafish genetics and vision and retinal degeneration / degeneration. That captures it.
Yeah.
Reasonably.
Yeah.
Remember the bio in real time. All right, but we should put up one of these predictions. Which one do you want to do?
What's the second? The second one. I don't know. Remember what it is, but
this is Beethoven's DNA.
yes, that looks like a good one.
That's a fun one you learned this summer. All right, we're talking about. I guess this goes back to that question of nature versus nurture that we've just talked about, but doing a very interesting historical example. So if you guys aren't following us, click the follow button. You'll get standard internet units that you can gamble on predictions. Predictions on Twitch are usually video game related. Will Jim beat 100,000 points in less than 5 minutes or whatever it might be? But on our stream, we make our guests do their own homework by writing you guys questions about science related to their expertise. So I'm going to pop up one of those questions right now, and depending on your degree of confidence of either of the answers, go ahead and predict them and spend those standard internet units. Those are useless outside the stream, but they do let you, I don't know, unlock emotes, make us take a drink, where you can request a factoid. So yeah, but also there's a lot of pride in answering these questions correctly. So we're going to pop up a question right now. So we sequenced Beethoven's DNA. It was through his hair. They had a hair sample, right? And so in sequencing that DNA, we learned some things about it. What did we learn? A predisposition for musicality or risk for liver disease? I love these because they're always like earwax or something, right? Like just something off the wall. But what do you guys think? What did we learn from Beethoven's hair from the DNA? I'm going to save that question. Contra, we'll get back to that one. Yeah, go ahead.
Oh, wait a minute. No, I just beat my high score.
Nice. Well, now you got to put Jim back in. Does it not?
Let's go.
Oh, we ran out of credits. Oh, there we go. Bunch of quarters. All right, so you guys have about a minute and a half. What did we learn from Beethoven's DNA? Predisposition for musicality or risk for liver disease? I love that the wrong answer, you can pick anything.
Yeah.
Just literally anything unless you guys have read the paper. But yeah, what do you guys think? What did we learn from Beethoven's DNA? Love that we got a chunk of hair.
And I remember seeing this paper myself, and then a colleague reminded me of it when he posted something somewhere about, oh my gosh, this is the year I'm going to add this to my lecture.
Oh, that's fun.
And I had seen that when your request for questions came in. I thought, well, that makes sense. Let's add that to the questions.
You never know where the inspiration comes from, but that's a really fun one. And you should add it to your lecture.
I should. I use an Abraham Lincoln in my genetics lecture. I use, we try to...
But only if you come in costume, right?
Yeah.
They got a piano on that stage, right?
I just shot myself.
Interesting. All right,
How
Jim,
we doing on the
what is the
there?
answer? Everyone said risk for liver disease. What's the correct answer?
Oh my gosh, everybody is correct.
No musicality.
Congratulations, no musicality. In fact, when it came to musicality, there's very few robust or well-controlled studies about genomics and genetics and musicality. But several studies out there can look at beat, how good are individuals at keeping beat,
interesting rhythm,
or
could be
tone,
some intrinsic clock biology related.
And it turned out that Beethoven scored in the lower 10 percentile in 2 of these. So he's not only did he not do well in the things that we typically attribute, he was really bad at things that we would attribute to musicians.
Wow.
So yeah, that's why I like that, or selected that answer.
Well, they've also shown that like perfect pitch is a learned trait, right? It's
Yeah.
not intrinsic in any way. Like pretty much anyone could learn it, assuming you don't have some intrinsic problem. Beethoven attempted to defy everything. He overcame adversity and became one of the all-time legends. That is amazing. Congratulations. But there was risk for liver disease. What did Beethoven die of? Do we know that?
He had liver disease.
Oh.
And, that was an era when individuals consumed far more alcohol to begin with.
Mm-hmm.
Let's see, I can't do anything.
Nurture versus nature in his case, apparently. In terms of learning musicality, I guess
and
that's the case.
So he had liver disease, and so this suggests that he was predisposed to the liver disease he had, and so the environment I believe he had hepatitis. They could at the time of his death, and so it all added into a harder or hardship for him at some point.
Yeah, I don't remember Beethoven's backstory. Was he a savant from the start, or he was...
I don't think he started as early as Mozart, but...
Yeah, because Mozart was like 3, but his sister was also very good. So that's another nature versus nurture question, I guess. Oh, with the next ship. Ouch. Man, that's... Yeah. All right, Contra said he himself related his deafness, or onset of deafness, to a serious illness with fever that hit him a few years prior. He said his gut never recovered, and he had severe lead poisoning.
Yeah.
Oof. Life back in the day was rough.
Yeah. We had inherited a set of pewter from back in the day, and we won't use it because we don't want to end up with lead poisoning.
Yeah.
So you never knew how much or little lead leached out of pewter.
Contra, his father, backstory on Beethoven. His father tried to make him into Mozart, but Beethoven had to take care of his 2 brothers because his mom died young, and the father was a hopelessly abusive alcoholic. Oof. So he overcame a lack of rhythm and that. So,
wow.
Good on Beethoven.
Wow, that's amazing.
All right, Contra has a question on your research. What have you discovered through the zebrafish? Zebrafish, what are they good for?
What are they good for?
Defend your zebrafish.
No. We have to defend the zebrafish. And this would be, let's see. So in the early days, when we didn't know many of the genes that caused human disease, we used our model and we did mutagenesis and looked for phenotypes that look like human disease. So we've identified several genes that were later found to cause eye disease in human populations.
Are you good on beer? Or you need
I'm good. So they were models of those diseases before, they gave clues as to where to look. Other things we do, most of what we do in my lab is basic science. So it's not going to make it to the clinic. We did receive several patents for some of the disease models that we generated. No company has seen them good enough to buy them up yet.
Disease models, as in selling it to other researchers?
So if companies wanted to screen those diseases, etc. And, the reason for the patent wasn't to get me famous or rich or anything. It's basically if the funding agency, it was a private foundation again.
Yeah.
And if there is any money to be had, then a portion of it goes back to them.
Oh, wow. Okay.
Actually, yeah, much more than I would ever see, which is perfectly fine. They were generous to support my lab.
That's part of the agreement. Anything you discover during that period.
That's right.
Yeah.
That's right. And that's fine. That is perfectly fine. We know that going in. And
I was going to say the American Chemical Society Petroleum Research Fund is the same way.
Yeah.
Like anything you try to patent as a result of the dollars given to you. Yep.
Right. Yep. Right. Yeah. It's an interesting system. And
all right, do you want to keep playing this? You want to do something else?
Switch.
We're 50 minutes in, so
yeah, we better switch.
We better. It's time.
Yeah, let's work on carpal tunnel on different figures.
What do you want to go with?
Oh, where's the list? So many choices.
So these like Simpsons and X-Men are just like walk and punch.
Yeah, I don't care for those so much. I don't know, maybe Centipede. Because that one just keeps like running. Yeah, is it on there?
So Centipede will go up. We'll get that guy to start. Sorry, we do a pause screen while we search through the games because we're playing this on totally legitimate emulators. All right. There we go. So same thing. Get you a bunch of quarters. This game's pretty brutal. I remember this one.
Whoops.
Not going?
Nope. Is it here?
No, it should be that one. Let me
Go ahead.
Rolls quick.
Was there another question on there? What might epigenetics not have?
Interesting.
Yeah, so basic science research, we try to find things like just how biology works. How do we develop is what most of my lab is. So how do we make those rods and cones? They have really interesting shapes, and hopefully the things that we discover other people can build off of.
I used to love the old rolly ball Centipede cabinets. Yeah. I don't have a trackball for this. We're doing it on joystick, unfortunately.
It's okay. Just makes it a little harder to control where the.
You can do. I think you got a little bit of up-down movement.
Yeah, we do.
Space. And yeah, we don't program in the sensitivity. Retrospect, we should set that up. But we're going through a lot of games. On the strategy of shooting paths, I remember this one. Let's see if there's any speedrun records on this one. Okay, so the question, we caught up on what have we discovered through zebrafish? Perfect.
But zebrafish get cancer. Zebrafish, one of the cool things we do with zebrafish is they actually regenerate tissue. So if they get damaged nervous system, they can grow new neurons. We can't do that. And that's
awesome. So Centipede, the game. So there's a bunch of different versions. There's like Dreamcast, Genesis, but if you look on the speedrun, there's extra life bonus. Get to that as fast as possible. Somebody does it in 32 seconds. There's a 6 live run. There's also a 999,999 points. How fast can you get to that? Let's see what the arcade record is.
Okay, I'm at a disadvantage here with the joystick, but it's okay.
Oh, man.
Normally, what instrument I have in my hand does not matter because I'm not good at gaming.
Wow. There's one time for getting to 999,999 points, and it's 1 hour and 17 minutes of this.
Wow.
So they got to
I think I knew that
as
guy.
quick as they can. Probably all knew a guy like that. But this is probably true arcade where they actually have a cabinet in their house. Man, that's really fun. Man, the trackball for these arcade games had to be a nightmare to maintain.
Yeah.
It's just like Doritos and Cheetos and whatnot.
And cigarettes.
Yeah, cigarettes.
That was the good old days. Remember cigarettes?
True. Oh man, have you seen this? The not just the vaping, but the Zims, like the tobacco pouches and stuff making a comeback. I'm like, we beat this shit in the 90s. Or we are again.
Yeah, once they start going to the dentist regularly, the dentist will put an end to it.
Yeah. Hopefully.
Got myself.
It's a veteran move, though. Pull them down. And I like that. Beethoven question. So this was a paper. So someone got a sample of Beethoven's hair.
Yes.
And so there's some IRB process associated with getting...
No, because it's probably a museum specimen. I don't know what family members are left that would actually control Beethoven stuff.
I see.
And so if it's archival... if there's, many times museums control what happens to that. Or I'm sure there's a museum that controls.
Interesting. So they just have Beethoven's hair lying around. So they sequence it. Oh my God.
Thought I could get
a nice move.
It didn't
so
cost
they
me.
sequence it and they just track it against known markers. And that's the process essentially.
Right. Yep.
So how do you feel about 23andMe has gone debunk, but have you done any of those popular sequencing?
I did. And the rationale is students would ask. So I thought, well, I should kind of do this, see what it's like.
Yeah.
You know, Google already knows everything about us, or Apple does. So what's the difference?
Man.
People would say, "Oh no, now they have your data." No, they have our data.
Yeah.
I'm not a conspiracy nut either. It's just think about it.
Well, my only concern with that is the health insurance, right? What defines a pre-existing condition? So, man.
Well, I'm older than you, so I have less time to worry about it. By the time we get around to that, then
The bright side of life. I like it. Man, that would be an interesting, like, let's do some machine learning, genetic sequence versus Google search history. Just see what pops out as a trend. Contra said Ancestry works quite well though. Whole genome analysis is down to like $300.
Yeah,
but that's not a whole sequence, right? That's major markers. Like, how does that work?
they may have reached the $300. I haven't paid attention to what Ancestry's doing
For a full sequence?
for now.
Holy shit.
It's pretty cheap.
So what the human genome was what? Early 2000?
Yeah. Wow. But you know, once that was done, and what the human genome had going for it was we had great genetics up to that point. So we could tie down new sequences pretty quickly when they were identified because we knew markers next to them. So assembly was made easier. But today, now that we know the human genome inside and out, it should be pretty cheap.
Oh, fun factoid for everyone. Human Genome Project, the first ones to solve a human genome, started in 1990 and completed in 2003.
Yep.
And that was Craig Venter. It was his own sequence, right?
That's the story.
You don't believe it?
So it actually, it started as a public endeavor, and then Ventnor peeled away and did his own private version. And it really became... that was wildly successful. They also sequenced model organisms. So mouse, worm, fruit fly, eventually zebrafish was added. Because what's the point of having, we don't do experiments on humans for important reasons.
Yeah.
So we use model organisms. And so we needed to know what was alike and what was different at the genome level. But it also allowed lots of biology. Genome sequencing is an engineering feat as much as a biology feat.
Yeah.
That was... But once we had one sequence, then it was easy to do many. Easier. And then we started finding out the very interesting things. Because when you have one, you don't know necessarily what's variation. And, human population, as a geneticist, it's the variation that's most interesting, not everything that's the same. So having lots of genome sequences. The coolest genome sequencing of recent is Neanderthal DNA. So we all carry little bits.
Oh man.
Okay. Whenever it's time for let's ask that one. Boy, what a lead in.
Yeah, no, that was a perfect transition
to my own question.
So I modified it a little bit. I just said which one.
Yeah.
I was going to do least or most, but either way, sequencing is hard on this particular option. All right, if you're not following us, click the follow button. We'll get the standard internet units that you can gamble on science questions that we're going to feed to you guys right now. So we're talking about genetics, in particular Neanderthal genetics that's laying around. Name is not compliant with our content guidelines. What? What's the negative statement? Pop? Wow, look at this. Put pop period. They were unhappy with it. Look at that.
Which were they unhappy with?
I don't know. Neanderthal?
Did I misspell Neanderthal?
Let's see if Neander...
A real wall. Word.
Wow. They will not let me ask a question that has the word Neanderthal in.
Really?
No. Which human population with the least Neander DNA? In this case, Neanderthal. Sorry, there it is. Neander is short for Neanderthal because I can't do the entire word. I don't know. I don't know what the rules are. I don't know what filter they have. But the question is, of these populations, predominantly Asian, European, or African, which human population has the least Neanderthal DNA? You guys will have to forgive me on the Neander. Wow.
That's interesting.
Man, I don't know. There's So I'm Mike Roperon, and he does microfluidics.
Yeah.
And that's an unacceptable word, presumably because of microdicks. But I don't know how their filter works, so we're learning things today, ladies and gentlemen. But yeah, if you guys have standard internet units, click that predict button. Depending on what you've learned from the stream or what you know about Neanderthal DNA, answer the question, best guess as to which of these populations has the least Neanderthal DNA. And then we will get back to the conversation regarding this one, which is kind of fun. All right. Man, I don't know how they do the filtering on that. That's pretty incredible, though. You guys have about 30 seconds left on this question. Man, I got your question. We will get back to that one in a little bit. Rule breaker. Schmonite, welcome back. Thank you for joining us. We learned something on... See, they'll let them put it in chat, but I can't do it in a prediction, and I don't know why.
That's interesting.
Huh. Let's try. That's too far. See, I can type it in chat. I don't know. That was crazy. We all learned something today. All right, Jim. What is the answer? Which human population has the least Neanderthal DNA?
African.
African.
But it's not zero. So Neanderthals left Africa, and then modern humans left Africa many, many millennia, centuries, tens of thousands of years later, million years later, maybe.
Wow. So we had 94% said Asian, only 6% said African. Congratulations on your 16-to-1 payout, everyone. Let's see whose return ratio. Very solid. Congratulations. Old man, good job. 10 standard internet units. You did Dark Horse Bet. Congratulations on it. I don't know if you knew that. If so, impressive. But that's not something you predict. I mean, if that's the cradle of life.
It is. And it makes sense if you think that, okay, everything arose from Africa, humanids. And then they came out in waves, the different humanoids. But all populations carry Neanderthal because, there's a lot of mixing in humans. One continent back to another continent, because we're talking over tens of thousands or hundreds of thousands of years. And, people don't stay put. Never have. And so there was lots of admixing. And initially it was thought that Africans had no Neanderthal DNA. But that was wrong, is because it was being removed. Because the standard that was being used was actually Neanderthal DNA. They were considering, well, it's not going to be here, so we'll remove anything that's not here. So they were removing it. The scientists were removing it computationally until somebody had an idea that, wait a minute, that is the right stuff.
Wow.
So that was pretty cool.
Huh.
So it was missed for 20 years until we changed how we were looking. So I thought that was interesting. But also, individual won the Nobel Prize for realizing that there's Neanderthal DNA in the human genome, because for some populations it's thought to improve certain immune cells and responses to certain pathogens.
Huh.
Right? So that's been carried over now for,
That was a very satisfying shot right there.
Yeah, lucky shot.
Just like, bop.
Thought I had that one. Wow, EJD, you're way up there.
Who's to say EJD?
Yeah.
Oh man, I wish it saved. I guess we have our NARC competition. That counts for something, right?
That's right.
Make guests play every one of these, but man, that's great. Congratulations again, Old Man Felinius that got the winner on that one, a 16-to-1 payout. Congratulations. That was a fun question. And so this is one. Going back to our discussion, or at least to the topic we're going to go revisit. So, your sabbatical. So, you took a sabbatical and in particular looked at human genetics.
No, I did more zebrafish genetics. I was in the human genetics department, but I didn't do human genetics. But I went to human genetics talks.
I see.
So, sabbatical, when you're... Ken has this available to him as well. But as faculty at many universities, after 6 years of service... then you get to go someplace, and for scientists, we usually travel somewhere to retool. And so you don't have your usual teaching duties or committee duties. But it's work. I went to work every day. But I worked in somebody else's lab to learn what they do so that I can bring that back to my place and teach my folks how to do the same thing. So I was back at the bench. At my stage of the career, I don't do that much bench work.
You were doing like actual wet work.
So I was at the bench.
Or what was the... Wow.
So some of the techniques we use in my lab, but the young people use them. I don't. So it was my turn to learn how. And then I do a lot of microscopy work, and my colleague does lots of microscopy work. And then one of the tools that zebrafish geneticists use across the globe, she developed. Kristen Kwan's her name. And so being in the midst of just where people develop certain tools and technologies is always a great feeling.
Sabbatical, though, I should just commit, right?
You just have to.
I mean, it's hard with the kids because you're trying to decide like the optimum time, and there isn't one.
That's right. So this was only our second sabbatical, and a lot of it was dictated because of kids, and then there's that pandemic thing.
That happens.
So it disrupted things. So like sabbatical. Some people take sabbatical at home. If they use it, they may write a book, a textbook for a class they've been teaching.
But at the very least, it's teaching and service release, right?
It's teaching and service release, and and it's not just scientists that do this. It's the arts, humanities, and but it's a way to go back and be the trainee again, is my view.
Yeah.
And
so you stuck in the domain of genetics and zebrafish genetics and new tools within that. But if you had to pick a sabbatical outside of your domain, like, I don't know, start over as a grad student kind of, what would you pick?
Oh gosh, I thought about structure at one time. So that was one of the options I was looking at. I do mostly molecular genetics, and there's now some really cool behavioral genetics out there. So that's still neurogenetics. I'm pretty happy with what I do.
Yeah.
But I'm like, oh my gosh, what would I do? I'd go back and learn how to write.
Like casual write, like actual literature and not scientific.
And if I could do anything, so I'd like to follow things in economics. And, I thought, well, maybe I should do a sabbatical in like economics. Apply some of the things that we learn in biology and see if it...
there's a lot of evolutionary models within economics.
There are. And it's just like there's certain, like social psychology came out of biology, and E.O. Wilson's study of ants. Now even a lot of social psychologists say that maybe that wasn't the best way to go about the whole field of social psychology, considering ourselves ants.
Framing. It's the spherical cowl of social psychology. Let's pretend we're all ants.
Yes.
Imagine you're an ant.
If society were ants,
how would they respond?
Well, it led to these office networks where people are pushed into either cramped quarters or they're given a certain amount of space because it all came out of ants. And I'm not sure people know this, but we are not ants.
Believe it or not.
Believe it or not, we are not the same as ants.
A little smarter, a little more.
But
less deterministic.
They are diligent. But no, it's also sabbatical. You're living in. I like doing it because you go live someplace else.
Yeah. And
in our training, we tend to move around a lot, and this is an opportunity to live someplace else. And whether it's another state, another country, city versus country, or rural versus urban.
Yeah.
And so just having a shift in lifestyle, so it breaks up the routine significantly, so it makes it easier to do other things.
Yeah. Well, just a full brain reset and consciousness and day-to-day life.
Yeah, and you start feeling good, and you agree to come on Ask a Scientist Gaming. Right?
Welcome back, Jim. When did you guys get back? Weeks ago, right?
No, we got back in the middle of July, but then I teach a course in Massachusetts in the summer, and so then I was home for just a few days and I was off.
Oh, interesting.
It's not bad. It's on Cape Cod. It's a long-standing marine lab called Marine Biological Laboratory.
Oh, that's fun.
It's been there for maybe 130 years.
Contra, we are a little less cooperative than ants for sure.
Yeah.
Man.
Yes.
Ant research is some of the coolest shit ever. I don't know if you guys have looked into this, but the way they figured out that the way ants figure out their distance from the nest, I don't know what the right descriptor is, but it has to do with the number of steps they take. I don't know if you're familiar with this.
No.
But the way they figured this out is they glued little stilts on some ants, and if the stilts were 20% longer, their distance traveled would be 20% longer. And so ants are just executing, here's the number of steps.
Wow.
My favorite ant study of all time is when ants die, like, how do they figure out an ant is dead, right? How do they decide we need to bring this ant to the graveyard? And it's not necessarily a conscious decision. And it turns out dead ants release something, it's like an oleic acid derivative or something like that. It's a molecule that dead ants, when they decompose, they give it off. And what these researchers did is they took an ant, covered it with this chemical, and sent it into the...
A live ant.
Yeah, exactly, and sent it into the anthill. And what ended up happening is these ants would pick it up, put it on the dead ant pile, and then walk away. And that ant would obviously get up and walk back through the catacombs. And then somebody else would pick up the ant, put it back in the graveyard. So, yeah, that's how they learned. I don't know what that was.
There we go.
Yeah, I love ant studies. Ants are, I mean, they got to be some of the most deterministic animals on Earth, right? That just execute an algorithm.
Yeah, the hymenopterans, bees, ants, wasps. They're great for study. There's wasps that have facial recognition, so that's how they tell which wasps are part of their nest, a form of paper wasp, I believe, and understand it. So they're computing in their tiny little brains little wasp faces. And you can do this computationally and make fake little faces to see what are the distinguishing marks that they're keeping track of. Now, do all wasps do that? No, it's a small clade of wasps that actually do facial recognition.
Wow.
Yeah, I think insects are really interesting. My favorite, being a vision person, is how there's ants that usually we think of insects, they stay out of the light when it's really bright and hot, but there's an ant that lives in a desert, and it comes out midday, and it orients using polarized vision.
Oh.
So it has specialized the way its eyes built, then it can tell where the sun is based on, or where the zenith is, because of the plane polarization that occurs through sunlight.
Wow.
It's a small amount of sunlight. Yeah, small amount of sunlight's polarized.
Yeah.
And they can detect that because of the way the cells are oriented. So they can do this cross comparison of just a few percent of polarized light to figure out where.
Are they seeing the sun directly or the bounce off the ground?
I think it's directly.
Wow.
Because it's a stripe that runs across their dorsal part of their eye.
They use anisotropy to figure out the time of day.
Yep. Yeah.
I did not know that existed. I got to write this down. I'm not going to remember it tomorrow, but that's amazing.
Yeah, we should have more to talk about because you do that photon stuff too.
Yeah, exactly. Well, we used like anisotropy measurements, right, for emission and absorption. So we try to figure out the orientation of molecules using polarized light. And we have a really cool surface bound measurement that does that.
Yeah, so that's interesting because fish, these photoreceptors, these cells that, when we make a nervous system or any part of any organ, there's the right number of cells in the right place at the right time. And in fish, the photoreceptors form this beautiful checkerboard or mosaic where the cells line up in beautiful rows, or some have a square pattern. And I would love to be able to figure out how that happens because, as a geneticist, if something's patterned, then it takes genes, it takes energy.
Yeah.
But, colleagues have tried to figure out it's not for polarization. We're not quite sure what they gain by putting these. If they had two cell types, then they would be just like a checkerboard. And some for photoreceptor cell types, so they see red, green, blue, and UV, they'll be in squares. So they all... there's a center one that's usually blue or UV, and then surrounding it are the other colors. So it literally looks like, squares around a center pin.
It's like a grid.
Yep. Yeah. And whenever, when biology doesn't do things by chance.
Yeah.
This frequently, right? Things can happen by chance, but not again and again and again.
And if it's not useful, it disappears.
It disappears because it's energy. It takes a lot of energy to build pattern.
Wow. That is really cool. All right, I'm going to catch up on some here. My partner said that they can recognize their own reflections. Presumably this is WASP's discussion. My partner said they can recognize their own reflection. Is that true? It's what a weird, yeah. What a weird science. What weird science might have determined that factoid? Man, that's a non-trivial chunk of animal research, though. It's literally putting a mirror in front of them, right? Like,
yeah, it's animals respond to mirrors. They try. What you have to do in many cases is grow these animals up in a naive setting, so they're not seeing other animals or they're not seeing themselves. And then many animals, they have a laterality, just like we do, right and left.
Yeah.
And so even trilobites, this is one of my favorite factoids.
All right.
An analysis of bite marks on trilobite fossils, and the bite marks are on the majority on one side of the carapace. And likely what occurs is during an attack by a predator, then they turn in the same direction. So then they're biting one side of the carapace. Right? And when you release an animal that's lived in a flat black container, even a little zebrafish, when it swims up to the first object that's reflective, it's seen itself. And then does it peer at itself with its right eye or does it peer at itself with its left eye?
That is amazing.
And so that's a form of laterality, just like we're left or right-handed.
Yeah.
Then that's laterality. And so that's an interesting concept in biology. We think of it, well, right brain, left brain. I mean, really, there's only brain.
Spoilers. That is one of the biggest myths in neurobiology, right?
Yeah,
left brain versus right brain.
Yeah, it's not like 100. It's like 49 to 51%, maybe in some people.
And if you cut out a portion, it'll just compensate.
You compensate really well. Yeah, that's what I think is incredible. So we don't regenerate new brain tissue, but the brain actually, especially in children, is quite plastic. So when a friend's child had a brain tumor, they had part of the lobe removed. 2 days later, when we took our son to play with her, they were back on the ground playing just like nothing ever happened.
Wow.
And and had their little trucks out and everything else. In the
I'm
hospital.
sure there's something subtle that like.
Otherwise, no, the kids are now 36.
Wow.
They're,
holy crap.
Within their 30s.
Man, that's really fun.
Yeah,
I love that trilobites have a handedness.
Trilobites, according to that, so that's the...
If you believe, yeah.
Yeah. If you believe that study, then trilobites have handedness.
There are some people with only 10% of normal brain matter, and many of them function just fine. There's some people with 100% brain matter that don't function fine. So, yeah, we run the
Define
spectrum.
fine.
Yeah, exactly. None of us are okay. Man, that's awesome. All right, I got to catch up on some. I'm coming back here. You said zebrafish regenerate neurons, brain or other? What neurons? They can generate all of them?
Almost everything where it's been looked at. So one of my favorite studies was done by a investigator, Ken Poss, when he was at Duke. Now he's in Wisconsin. But Ken demonstrated when he was a postdoc that you could snip off about a third of the heart ventricle. So that's the major pumping. Fish have a little different heart than ours, but it essentially just pumps the blood like ours. And you can clip off a third of the ventricle, and as long as you don't damage the heart so much they bleed out, then they grow back that missing third. Our problem is when we damage things like our heart, we form scar tissue, and that remains a weakness, or we never recover from that large amount of scar tissue.
So they don't generate the scar tissue, or they clean it out?
Very little.
Wow.
So it's a lack of generation of that. And then another colleague who was in Ken's lab, Maina, she demonstrated that she could go in with a fine jeweler's scissors and snip the spinal cord. And so she could cause a spinal cord injury that severed the spinal cord. And lampreys will do this too. And the mechanism is a little different. but the fish will lay on the bottom of the tank while the axons regrow, the motor neurons, and then reinnervate the muscles of the fish, and then they can swim again.
Wow.
Lampreys do it a little differently. It's that there'll still be a little cleft or scar, but then the neurons kind of reroute and get to the muscles, and so then they regain function, and there's some learning involved. So they may not regain 100% of the neurons, but they regain 80% of function, so they can swim again, eat, hunt, etc. Because then they adapt. So they kind of retrain the movement so that they get their beautiful lamprey swimming motion back again.
That's amazing.
That is amazing.
So what would, and you might not know this answer, but I'm curious, what's the evolutionary advantage of scar tissue then?
Wow, yeah.
It's useful, presumably.
Yeah, those of us that do a little regeneration in the lab on the retina, and primarily to understand if the genes in regeneration are the same that we study in development. In some cases, they do the same thing. In some cases, they kind of swap with other genes, which I think is interesting. So it may be that we're less adept at swapping out genes that were shut off, and these animals can turn them back on. Typically, they're in regenerating brain tissue or retinal tissue in zebrafish. There's populations of cells that are support cells for neurons. Well, they can make stem cells. But when the heart regenerates, just saw Ken a few weeks ago at this course I teach, and his lab demonstrated that the heart cells actually make the stem cell. So we don't do that. So the evolutionary advantage, yeah, this is something that we talk about over a beer. It's hard to prove what happened or why things happened.
Yeah.
We can only say what happened, and then kind of deduce, then speculate.
Yeah.
And it was one speculation is that we're long-lived, and you see more regeneration either in young animals or in short-lived animals. And since they may not be prone to cancers, et cetera, that you see in long-lived animals, or since we have less risk, or we have our organ systems are so dedicated to their job in long-lived animals that they can't reactivate these other programs. So we start seeing mitosis, cell divisions, but then it turns into scars because they can't make new neurons. And so when you make more cells and you can't do anything, it's just like a scar on your arm. It doesn't mesh. The skin doesn't match precisely. So the extra stuff, it does a good job at filling the void. Right. And so but these are debates that we have beers in hand. Yeah. So these are discussions that we have with beers in hand.
The conversations are fun.
These are, so can we, from a biomedical standpoint, can we trick our cells into behaving like a fish cell
without giving us cancer,
or to at last repair or regenerate enough, and then we can use training, we can use physical therapy, things like that, to actually regain use.
I mean, there's this the extreme example of this is like regrowth of a tail, right, of certain lizard species. The idea of, can you activate that in a human and regrow a finger or an arm or...
Yeah, there's the wayward reports of during delivery, children getting tips of fingers lost to forceps or surgical instruments, and then they grow back. Yeah, because as children, as infants, newborns, we're still making neurons, we're still making cells of the eye. Our vision is very poor, and and we also have use type accommodations where as we use our vision, then it starts mapping appropriately to the brain. And if we miss that critical period, then, in my day, kids that had monocular vision or lazy eye, in some cases, they put a patch over the strong eye to force the lazy eye to be used. But then they weren't getting the appropriate connections to the brain. So they'd end up with like monocular vision when they were older. And so, it was, I don't think we patch many kids' eyes like for this reason.
I was going to say, the question, unlimited budget, no moral qualms. How much of a kid figure can you cut off before it doesn't grow?
I'm sure it's in the literature. We don't really have to do the experiment. I'm sure it's been reported as some case study over what growth has happened, because a lot of kids get born. Yeah. And in many cases, and in a lot of cases, by accident, but you never know the circumstances.
That's crazy.
You know.
All right, we'll go start again. All right.
Yeah,
Contra, follow up in that conversation. Is there an age cutoff for human retinal regeneration? Can this be stimulated by stem cells?
Yeah, I read just earlier this week that there's a clinical trial, and so they're trying to recruit individuals. I didn't read it that closely because I was getting ready to leave work that day, and where they're looking for volunteers who would be eligible, but it's a phase 1. So this would be, because we do not regenerate retina, we do not regenerate neurons, period. That it just does. Well, okay, I shouldn't say period. There's small areas of the brain, part of our olfactory system, where we grow new neurons. Parts of the brain, there's stem cells in one region, and then we get a little bit of plasticity in another because we add new neurons. But for the most part, we're done by the time we're 5 or 6 years old, and then we're slowly declining from there.
You peaked at 5.
You peaked at 5.
Oh,
yes.
No. Oh no.
Yeah. So yeah, we don't regenerate nervous tissue. And the other constraint we have for, say, spinal cord injuries is when you sever an axon. So that's what connects the cell to its target, the neuron to its target. Like for twitching a finger, that axon goes from my spinal cord all the way out to the tip of my finger. Well, that axon grew when I was, a tiny little 10 or 12 millimeter bean.
That's crazy.
And now I'm not, a centimeter long or 5 centimeters long anymore. Axons have a limited growth rate. And so, what took months in utero takes years if we were to make those connections again de novo.
Interesting.
Now we're long lived, so we can wait years.
Problem.
Yeah, it is a scale problem, right? Wow. And it's a log scale problem. I don't know much about math, but log is big. Well, that's why I'm a biologist. I didn't understand math.
Gets big, gets bigger.
Yeah, gets bigger faster.
Oh, wow. That is interesting. And, and then thinking about that from a thermodynamic perspective, growing a millimeter is way different than growing an inch.
About a millimeter a day.
Yeah, that is intense.
Yeah.
All right, Contra, at what stage of life could you / would you apply one of the CRISPR treatments that would be based on your models?
In fish or humans? Ooh, okay, so if we're... So when we do this work in model organisms, then we're usually working on a fertilized egg or the first stage of the embryo, so that the entire embryo is that genetic, has a high probability of that genetic change. But for disease treatments, okay, so we have the unethical and the ethical. The ethical is when these are being used now for sickle cell cure. Yeah. That's I'm not sure it's out of clinical trials yet, but of course we're doing this on fully grown individuals. In clinical trials, we tend not to do things on children unless it is a treatment for children.
And even that, the restriction is...
The restrictions are, the bar is very high.
Last resort.
Yes.
Yeah.
Yes. And so some of these would be for adults specifically. And then others would be for children, hopefully. And then there's the study where a group in China deleted the HIV receptor from 2 in vitro fertilizations, and then those were implanted into a woman, and children were born. And this was done without any oversight by any committee. And they could do it, right? This isn't beyond what we can do in science. But the question becomes, should it be done?
Yeah.
Now, we don't have a good understanding of what the HIV receptor's function is in normal cells. But since it exists and it persists for all these years, genes are very easy to get rid of, right? There's no selective pressure to keep a gene that isn't used anymore. And we accrue lots and lots of mutations every generation. Mm-hmm. So genes get silenced pretty quickly. But we have this gene. And so it must do something besides sit there evolutionarily waiting for a virus called HIV so that it can infect us and cause a disease.
It's coming.
That's right. So we're going to hold out until we're the receptor for disease that's going to wipe out individuals.
Huh.
And so, but they deleted this in 2 individuals without oversight. And just because we can do things doesn't mean we should do things.
Yeah. It's the Jurassic Park conundrum.
Yes.
All right.
But wouldn't it be cool to have a dinosaur?
Exactly. Just build a good gate, you know?
That's
more than one.
Which one do you want?
Just make them very small, like chickens.
Chickens with teeth.
Exactly. Talons.
Big sharp teeth.
Old man Felinius falling back on the WASP and facial recognition discussion. WASP can do facial recognition with surprisingly few resources. I mean, when you talk about the number of neurons involved, it's presumably
that's what's in. Yeah.
But Microsoft Word takes gigabytes of memory to install and run and bring most CPUs to their knees. We have much to learn from more efficient systems. I mean, that's kind of a disingenuous comparison, though, right? Like, if you specialize in one task, it's very easy to do that one task efficiently.
Well, they still do all the other tasks that wasps have to do, like fly and find food and find a mate and be able to raise their young and hunt.
But can it move a figure around that I don't want it to go there?
Could we train it to do that? Because
exactly.
We didn't know we could do that till we had Photoshop.
Exactly.
We didn't know we needed to do that till we had Photoshop.
Oh man, that's a good question. Efficiency is something we're not good at.
No.
Man, even if AI is peaked, if we can make it more energetically efficient, it's super useful. What's your opinion on the AI? I'll give you my diatribe. It is a bubble that is going to pop really hard, and there's diminishing returns on getting 1% better for 20% more energy. I don't. Got to figure out better ways to do it, and it's going to be mostly specialized and not general models that are useful. But
okay, so are you done?
Yep.
I have my conspiracy theorist version.
Oh, let's hear it.
And that's that if we called it artificial intelligence, then people would be scared.
Yeah.
But I think that calling it AI was a marketing tool.
It's clever.
It's clever. And, so when ChatGPT first, I'm not plugging them, first hit the scene, and academics like myself were like, oh my gosh, the sky's falling. And I made a quick decision that I'm going to have my students all get an account and I'm going to teach them just how horrible it is. Mm-hmm. And then they would actually write papers on their own. And it worked.
Yeah.
And it's a couple iterations later now. And it's fun to use because I'm not a great coder. And if I need a fast Fourier transform,
yeah,
I just don't write those off the top of my head.
Yeah.
So when we needed one of those in the lab, the student said, well, I'll just ask chat. It's like, fine, and see if it actually works and does what we need. If not, then, you're bright enough to modify it in ways that we just need to see if that's the way we should go to analyze data.
Yeah.
And it was a lot easier just having chat write something based upon what our data were and the type of analysis we needed. And it was great. But for doing real original work, one, it's stealing, and number two, it's not really that original for most things.
Yeah.
Even for code, it's finding somebody who wrote and posted an FFT that worked. So it's not de novo writing us an FFT program for doing just some type of analysis. It's stitching together known things, and they tend to work. Where not all the sentences they put together or the scientific reports for individuals who won't be named. So it's really good at, because it can't tell the difference at this stage between real information and made up information. So it weights everything equally. So as we have more false information on the internet, and now there's one version that we were playing with, and it uses as its primary source Wikipedia.
That's pretty good.
So Wikipedia is usually a good first pass for things.
Yeah. Crowded. Yeah, there's a bunch of different large language models. One that I really like, I don't remember the name of it, but it exclusively uses peer-reviewed publications as that source information. I don't know how it gets that information, but it exclusively uses that, so probably more believable.
And the subscription version of chat would use more updated sources, where the freeware limits what sources it's using. And in those cases, then a colleague tells the students that they have to have sources after that date. She doesn't say why. She doesn't tell them that chat or whichever, I think it's a version of chat. It only uses sources up to, say, 2000 or 2020. So she tells them that half your sources have to come after this date. They have to be new. And this is the definition of new. And then she can tell people who are using chat because their sources are all old and they don't go back to fix them.
Oh man! So we had Tommy Usick on, he's in the linguistics department at FSU, and he studies AI.
Yeah.
And then one of the things I learned from him is that I don't know if you remember this phase of jokes where it was like, anytime the answer or text included delve,
Yeah.
it was ChatGPT. But it turns out delve wasn't a byproduct of delve in the literature, like the prevalence of use of delve. What it was is feedback from humans that said this sentence was better than this one. And people might have thought delve sounded better, and that's why it became more prevalent in use. And so it was the human retraining that was actually making that happen.
Oh, that's interesting.
Yeah. So that was a byproduct of humans and not literature, because he went through the libraries of data and said, here's the prevalence of use of delve and other words. And so it was humans actually causing that, which is pretty amazing.
That is. So it is listening.
Yeah. No, it takes.
But it's listening to us, which is the problem.
Yeah, we're terrible.
And the problem is we're the only ones that admit it.
Yep. Yeah. And so our bias in the training, and we're feeding, training it through our feeding. Contra said, Doc's neuroplasticity is on point. Watching him get better at these games in real time is impressive. Shout out for gaming. Look at that. Less mediocre gameplay. No, I think you went pretty far in that round. I don't know what level you got to. That was a solid run.
Yeah, it was okay. I never played many games. It was just like anything else, right? If it's there, you play them.
Oh, yeah.
But I didn't like plugging my quarters into a machine.
Oh, my God. So expensive.
Then I do stupid things like that.
I challenge someone to watch this YouTube video and see how much money Jim spent, assuming a quarter per life. Or a quarter per 3 lives. Yeah. I do love modern arcades where it's just like paid for 3 hours of play.
Yeah.
That's so much fun. Bringing the daughters in there and we'll just go to town. Old Man Filoni is following up on the discussion, the comparison of the WASP versus Microsoft Word. Absolutely disingenuous comparison. My point was more in the sense that we have much to learn from organisms that do tasks efficiently. Absolutely, there's more than one way to optimize a problem. I visited a biophysics lab in Nova Scotia and was blown away at the work they were doing and how biological nanomachines were assembled and function.
Yeah. Viruses were the original nanomachines. They're just a machine. They're not alive.
Yeah.
They're programmed to do a task, and they do that task extraordinarily well. But as we saw with COVID, certain viruses are prone to error, right? So that's why they mutated. The vaccine had only a lifespan of so long for really protecting you from death, but it's still pretty good at keeping you alive. Because they're so prone to error, that's kind of the strategy, right? Great machine, great at doing one thing, but they're prone to error. But then that makes more little viruses that do different things. So from a machine standpoint, I mean, all the biology is just a whole bunch of machines.
I was going to say, like protein, DNA, like all of its nanomachinery. Right?
Yeah, it's really incredible. And we sit here and we waste all that playing games.
How dare you, sir? The optimum use of the human condition.
It's amazing.
Video games and science are the pinnacle of achievement. Someone fight me. All right. Contra wants to know, do zebrafish have the same retinal pancreas connection and to the eyes that humans do?
Retinal pancreas connection.
Retinal pancreas connection. I do not know what that is. You ever heard that before?
No. Ask chat.
The AI overview by Google. Retinol as vitamin A is essential for normal pancreatic development and function. Retinoids are stored and metabolized in pancreatic stellate cells,
Yeah.
stellate cells, and influence pancreatic fibrosis, while low retinoid levels are associated with pancreatic cancer. So it shares material, right? Is that what that's saying?
Yeah, so development, lots of tissues use retinoids for development. We have what are called retinoid response element. It acts like steroid hormone receptors. And, you know, we think of steroids as testosterone, estrogens, or progesterones. And, but during development, these are really important for lots of signaling and lots of developmental things. And so in the pancreas, yes. The eye, yes, but then in the eye, we also use retinoids. That's part of the visual pigment. That's an interesting process by which the retinoids from carrots are the precursor molecule, but that makes the pigment. You know, pigment's a protein and that retinol. And so when photon hits that activated, that pigment that has the retinol, then it becomes activated, and that triggers vision. Right, so it's kind of two different stages. One is the developmental stage where it's triggering certain genes during development, just like other steroid hormones. And then there's the specific process in vision where it's used in a very precise signaling molecule. We call it visual opsin is the old term. George Wald won the Nobel Prize for his studies on visual opsin, and then why vitamin A deficiency causes vision loss.
Interesting.
And so that things like the orange corn or orange rice, so rice that carries lots of carotenoids, because one of the leading causes of vision deficiency in less developed countries is vitamin A deficiency. So there's a link. It's the same molecule, the same base molecule, and it's in biological time. One is developmental time, and the other is actually in the visual process itself. But wow, that was a good one.
That's fun. We dove deep on that. All right. You want to keep playing? You want to try something else?
Yeah, we could try something else.
What do you
When
want?
they notice that I'm getting better at it,
It's time to get worse. We can do this. We have the Star Wars game. We have
yeah. How about Star Wars? Because that's kind of a break.
yeah.
There's not much to do with that game,
Point the mouse.
and it's a mouse.
yeah. Visually looks cool.
So since I was working in a lab for the last 6 or 7 months, then I had to use muscles that I hadn't used in a long time.
You got sore.
And I got sore. Yeah. So you know, working the lab I work in, types of labs, it's a lot of fine motor control, but we do things slowly. So it's not gaming. Gaming is fast relative to what we do.
Yeah.
And yeah, so this thing called carpal tunnel syndrome, you know, it's a just waiting for robots to take over, and that would be fine.
All right. Start on. Yeah, that's the one we did earlier.
Yeah.
All right, ladies and gentlemen, we're playing some Star Wars. I can't remember who played this. I think it was Paul Peluso several weeks ago, but it was a fun game, and Jim was looking for a chill game just to shoot a whole bunch of stuff. I think you can hold the shoot, so you don't have to.
Oh, that's right. I need
have to-
reminders.
Yeah. Well, we don't want you to overstress your finger. So we believe it or not, we make a living typing at a computer, so we have to be able to do that efficiently as possible, like wasps recognizing a face. Lori, welcome. Thank you for joining us. Our guest today is Dr. Jim Fadool, and today we are drinking this is Guinness Draft Stout, and this is the nitrogenated. I don't know if this has the nitrogen capsule in it or they just seal it under nitrogen, but it's nitrogenated for smoothness, which is fun. And this is what we drank last time, right?
Yeah, I believe so.
Yeah.
I'm not particular.
Yeah. No, except you're particular about one. What was it? It was Miller.
Miller Highlight.
He was like, I'll drink anything except this one.
And that's a family thing.
Yeah.
It's yeah. It wasn't in Dad's fridge.
that was something. That was nurture
Yeah, that was yeah.
or nature. We don't know.
We don't know, but you know, I never grew accustomed to it. I'm sure I had it at some time in my life. But when you have a choice,
Yeah.
and I do have another one. I don't like high APV average.
Oh, I don't remember.
Yeah, well, the high alcohol beers because then they taste like alcohol.
True.
And if I want that, then I'll drink like whiskey.
Old man Felonius. I'm currently drinking Galaxy Starfighter Sparkle Puff Defender of the Universe IPA. That is amazing. That's what I love about all these, small local breweries, because they just make anything. Right?
Yeah,
they can name it anything.
And they can try new things. Yeah, it's really creative.
That's fun, Lori. I visited the Guinness Brewery in 2001. My entry ticket was a plastic oval with a half ounce of Guinness in it, and I still have it.
I did not take the Guinness tour when I was in Ireland the last time, but we took a bus tour that was promoted by Guinness, and there's an artificial lake that's in the cut of the.
I think left click.
Yeah. There's a lake, and that looks like a Guinness glass, and they even poured sand to look like the foam on the glass, and so that's the only thing I remember. I was over in the UK for some reason, then we zipped over to Ireland, and we were very tired, and we're not people that normally take many bus tours, but it was just right. The bus driver was a phenomenal individual.
That's awesome.
That told great stories, and so it was a lot of fun. Then you got to stop at this thing to take a little walk around the lake.
Huh.
And it was I can't remember if it was part of the conference that we were attending or it's just something we found at the last minute because we were so tired.
All right, 9:45. We should do another prediction. We have several here, and I've not been keeping on top of them. Which one do you want to do?
Let's see.
We can do the first one. We're talking about
beer.
Might
We're
as well.
talking about beer.
That fits. This one is reused. If anyone was here 4 years ago, you have the inside scoop. But if you don't, if you weren't here, well, then you're on your own. Can't remember the name of those guys. They smell just as bad on the inside.
Yeah.
All right, we got another question popping up on the screen. If you're not following, click the follow button, get your standard internet units, and you are going to answer this question without the help of ChatGPT or the internet or whatever it might be. What was the first recorded cultivation of hops for beer? Was it 753 BC or 736 AD? That's fun. Cultivation of hops for beer. It's better living through biology, chemistry.
Yeah.
Well, both. All right,
Flavor science.
You want another one?
I could use another.
I think these are canned with the nitrogen. And for those of you guys not familiar, like nitrogen is not very soluble in water, and so you have to pressurize it heavily, but it gives it a unique taste, right? Versus carbonated or whatever beverages might have.
I think it adds a different mouthfeel.
Yeah.
And that influences taste, whether things are hot, cold. Right.
There we go. Yeah, right click on that. That gives you a yes. All right, you guys have about a minute to answer this question. First recorded cultivation of hops for beer. Note, it could have happened earlier, but this is the first recorded, demonstrable version of that. So yeah, this one I don't know.
This is the foodie in me.
Oh yeah, we talk food and beer. Is that pretty light?
There were other
4%. Yeah.
Yeah, they're only like 4.2. So in Utah, first time I was there, alcohol purchases were very restrictive. Now you can only get draft beer in 5% or less.
Huh.
And you can buy canned beer and seltzers and all that at the grocery store. But again, they have to be 5% or less.
But they've lowered their restrictions.
Lowered the restrictions. If you want beers that are made across the country that are higher than that 5%, then you have to go to an state-run alcohol store.
Huh. And restaurants, they serve below 5%?
They can serve, bars and restaurants can serve cans that are higher than 5%.
Huh.
Cans and bottles, but anything bought from a grocery store, etc., or anything draft has to be 5% or less. So, which is fine. There's creative breweries out there trying to get flavor into these things, but it's which I was enjoyable to try these different beers. What I didn't like were, or what I found less enjoyable, were those that try to mimic higher alcohol beer.
They want all the flavor of my alcohol.
Yeah, but alcohol's it's a solvent. So some things go into alcohol that don't go into water well.
That's a good point. Huh. Yeah, so I interviewed at University of Utah, actually. Did not end up going there, ended up at FSU, but it was interesting going to the restaurant, and they had like lock boxes where the departments had their own selection or something. There's weird rules about what you could do and what you could bring.
Yep. So there were, and I think at the clubs you can store, they have these lock boxes. And I've seen these in other places, and our first position was at Auburn, and so you could buy alcohol at clubs on Sundays. And the club, you could buy a that day pass.
Huh.
So it wasn't a great restriction.
All right, Jim, what is the answer? First recorded cultivation of hops for beer.
AD.
6 AD. So
Yes.
Fairly recent invention in the spectrum of drink.
Drink and food, yeah. So 753. So I looked up 700s to see BC, or before current, when what were people drinking back then? And of course, mead. So there were fermented grains, but mead was popular. And so fermented honey water with different flavorings. And then there's a resurgence in mead. So that went down the rabbit hole of mead for a while, and how, mead and ciders were more popular than beer in most places until at least these two articles, which may have used the same source, until sugarcane production grew. And so then honey was lost its pillar or stance on the pillar of sweeteners. And so then with grains and sugar and molasses, then other items became the favorite for beverages.
That's fun.
And fermentation. But mead's interesting. Again, a lot of craft meads out there again. So
next time you're on meads,
so try them. They could be interesting.
yeah. No,
Actually,
we've had mead on stream. I don't remember who. It's from computer science. No, scientific computer.
there's a small town west of here, Apalachicola, who was known for its oysters, and they have a place that produces mead now. And one of the breweries that was there has moved to Tallahassee, Oyster City. But they have another brewery there. But the mead, I was just there the other day. We try to get there once a summer. Quiet little town. And,
don't remember who picked mead, but yeah, you can buy it at Total Wine. It's very sweet though. It's a shock to the system.
It can be, and actually it can be a much higher alcohol content because then they're really controlling how much sugar is in. They're not limited by the wort from the grain. So they can really push the alcohol content way up. So some can be fizzy, some are flat or still.
It's a lot of faces. I'm not going to be able to find it. So it goes.
The mead?
Yeah. Oh no.
Oh no.
Did we hit no? Well, if you move the mouse right, it might have gone to the no option.
Okay. I'm still learning. I didn't like the Endor. Too many trees.
My favorite factoid about this question, actually, is we asked this 4 years ago.
Yeah.
And this still holds the record for the biggest payout ever at 256 to 1.
Really?
Yep.
How do we do today?
It was only a 2.36 to 1 payout. Well, 1.73 for the winner. So yeah, no, this was much more balanced than the last time we asked.
Yeah, people got they're either maybe that last group was not as well informed as this group.
Or they were overly confident. Locked on the wrong answer. That's what I love about this. It's not just who's right and who's wrong, it's how much you bet on it. How many internet units you put towards a given answer.
Well, that's it. I'm glad I reused the same question, especially that question, if it holds a record.
Very interesting about the hops. Also, Apalachicola is a cool spot.
Yes, it is. It is. There's lots of little towns all over the place that are really cool. But we like that one. Interesting history.
Mead can hit like a ton of bricks. I mean, it's deceptive, right? Because it's like a sugary drink, but you can still get like 12% alcohol.
Yeah, yeah. They push them up there. It's essentially the same as wines.
All right, if anyone has questions, we're about to cross the 10 o'clock line. I think at, 10:30, we'll probably switch over to NARC, so we got another...
Oh, gosh. I'm going for the record this time.
Are you?
I don't even make the list.
So this, about 36 minutes left. So if you guys have any questions for Jim, our guest today is Dr. Jim Fadool. He's an expert in neurobiology. He uses zebrafish as a model to study genetics, particularly as related to vision and retinal degeneration and regeneration and how that occurs. So if you guys have any questions related to that, or biology in general, or genetics, we are happy to discuss them. We've been down quite the rabbit hole tonight of questions, so we are happy to discuss as things come up. All right, so one of the canned questions. Relation to D. Fadool?
Yes. It's not a common name.
Exactly. Not a coincidence, but yes, Lori was our former graduate advisor in the chemistry department, so she would come across the name D. Fadool many times. But yes, Debbie is Jim's wife, and they were both just on sabbatical for the last year.
Yes.
It's kind of fun. So you guys got separate labs or same lab?
We were in separate labs. She was in a lab doing calcium imaging. She's a physiologist or biophysicist, channel jock, as we say. And so she wanted to try a different type of physiology recordings. And this is where transgenic animals carry a dye that fluoresces, and the fluorescence changes whether there's more or less calcium in the cell.
The color of it changes?
The intensity.
Interesting.
And so it's a genetically encoded sensor for calcium. And so then as the brain is doing its thing, then the neurons are flashing brighter or more dimly. And then you have to decode this using, programs that are way beyond my ability to write.
That is intriguing, because intensity is a really hard thing to measure.
It is, because they're always flickering.
Yeah.
And so, it's how much change was there over the baseline. And so it requires, and the change is very small, just a few percent change. And then you have to subtract out the noise, of course. And you're trying to track hundreds or thousands of cells.
Oh.
Whoops, there we go.
There we go. Oh, no. Time ran out. Oh, my question for Debbie would be, what about measuring the emission lifetime? So lifetimes are easier because intensity can change with a lot of things, but the lifetime is intrinsic to the environment.
Right. And so, I'm not sure there's any lifetime. Now that I think about it, there are lifetime genetically encoded probes, but they're not used as much because they're newer, and there must be some limit to the lifetime. Because I know when we use chemical probes, then lifetime is actually when we're trying to say two things are close together in space, then lifetime is better than just using total fluorescence or fluorescence energy transfer, things like that.
Yeah.
So the change in lifetime for the donor is what we track.
Oh, that's for like an energy transfer quenching thing? But even for the stuff that Debbie was measuring, sorry, I need to talk her into being on. She said no, though. You are, right? But it would be fun to have
She
her
doesn't
on,
drink beer either.
Because the reason the intensity changes is because something with the decay is changing, how fast it decays in the ground state. It should manifest in the lifetime.
Right. And so when it's bound to calcium, then that's what's changing, is my understanding.
That's fun.
But these probes, there's dozens of them out there since their first discovery. People have been manipulating them to make higher efficiency probes sensitive to different ions or compounds.
That's fun.
Probes that are faster.
Yeah.
Right? Because imaging is a rather slow way to do biology.
Yeah.
We work on the order of tens of milliseconds, and biology in many cases works on, much faster scales, order of 10 or orders of 100 faster. So you're getting a response that's representative of something, but it may not be the actual signal.
Mm-hmm.
Right? So it's where what she was dealing with before on these orders of, microseconds or tens of nanoseconds were the actual biological molecule moving.
That's so cool.
Right? Opening and closing, as they say. So it was a more representative, but then, of course, you're catching narrower bits of data.
Mm-hmm. And that's so cool.
Yeah. It's kind of like what you do, but on biology things.
Yeah, well, that's one of the things I've always wrestled with. Like, I've published in pretty much every domain, analytical, materials, inorganic, physical, organic chemistry. The one I haven't... I have one paper in peptide science, believe it or not, and it was using this synthetic peptide to put a molecule on a surface for electron transfer for solar cells. So it's very much cheating. But I'm on a draft right now with Ryan Sturms. Who was actually a guest on stream when he visited. He came and used one of our spectrometers, and he was looking at oxygen binding and dissociation, because there's hemoglobins across, like, not just animal life but also plant life.
Yeah.
And it's a question of what those hemoglobin are for. Most of the time it's not oxygen transport, right? It's some kind of redox chemistry. And so he's basically looking at, the binding rate and whether it's, 6 coordinate or 5 coordinate. And so, yeah, he's going to submit it to biochemistry. So that's my gateway.
Those plantains are what give the fake meats their meat-like consistency or flavor.
Yeah.
The hemes.
And it's the same structure. It's just doing a different job in these different systems.
That's right. But it's also so these actually came from symbiotic with a plant.
Huh.
The ones that are used in the meat products or the fake meat products that we buy. And but these nodules, if you think nodules on pea plants, if you ever grew pea plants.
Yeah.
But the nodules in these are pink because of the heme binding capacity. Huh. So blood's red, these nodules are pink.
Yeah.
And I think it's cool because I like biotechnology type things.
Yeah, I was going to say that's a cultivating strategy, and symbiotic, or that's just a coincidence?
It was a natural a natural occurrence in the environment, right? So some plants have nitrogen fixing bacteria with them. They're associated with, and us too, they're symbionts. Our whole gut lining full of bacterium. They're not they're living inside us as an organism. They're not inside our cells, but there's some mutualism, I guess would be the better term, symbiont in that case.
That's fun.
Yeah, but cool things like that, and then you think, gosh, why didn't I think of that? I used to do
just the stupidest simple ones that, like, that makes sense.
Yeah, I used to do tool building at one time, and so when I see other things like talons, then CRISPRs, and I'm amazed that people come up with such cool things.
Yeah.
Right. It's
that's fun.
Hit the event button to shoot photon torpedoes.
That's the right click. So hit that target.
I missed.
No, the Death Star lives on.
I knew I missed.
Sorry, Alderaan. I let you down.
Oh well, it's not the first time.
So many electrons lost. Oh no. But yeah, I keep saying, like, I wish I would get forced to teach biochemistry, because that's when I'd actually learn it. When I had to.
Yeah, no. I took a lot of biochemistry classes.
Not worth it.
And it's the biochemistry that gets taught. The interesting thing, it's enzyme kinetics. There's topics that are cool.
Yeah.
Inborn errors of metabolism, but the way it's taught, I don't know if it has to be taught this way. I don't teach biochemistry, but there's a lot of just memorization.
Yeah.
Right. There's lots of things to learn just so that you can learn biochemistry, and that wouldn't be fun to teach because the students don't like to learn things like that. But it's like learning a new language.
Yeah.
And you can't have a conversation until you learn that language.
Yeah, I mean, just starting with the language, the acronyms, like...
Yeah. There's the giant wall chart of biochemistry. Because you have to teach everybody from energy to lipids and you know, I kind of skipped lipids.
Who needs?
It turns out, yeah, there's some really interesting biology in those there lipids.
Steve Leonard likes them.
Yeah. You know, we build membranes out of them. We do some
Yeah,
cool
exactly.
things with
What are
lipids.
those?
It's
One
not
thing-
just
One thing Steve brought to my attention that I really appreciated about lipids was that we can reproduce most things in cells, right? Like DNA and proteins and stuff.
Yeah.
It's really the compartmentalization that makes it effective. And I did not appreciate that fully until he brought up that point.
with the lipids.
Yeah.
Yeah, that's one of my early cell biology classes as an undergraduate. That was kind of the takeaway message of a cell. The coolest thing about this is we always just thought, you know, all the extra space in between all these lipid compartments was just like stuff floating around in water. I guess that's not quite what we thought, but for the most part, that's what we taught.
Yeah.
And it turns out it's not even that. There's even the space in between the different organelles that are lipid bound. They're regulated in what's in there and what's not, right? So there's domains within this, what we thought of as a water-filled domain. And so things are, even without the lipids, there's regulation. Because bacteria have no lipids on the inside of their cells.
Oh,
It's
interesting.
compartmentalization, but they compartmentalize too. We compartmentalize bits of membrane with protein.
I did not
So-
know that.
Yeah, that's the cool stuff. Oh yeah, you'd want to teach in biochemistry,
Yeah.
but they don't know enough biology or chemistry to actually, I think, appreciate it. But what do you think? Could we just jump in? I mean, I taught my oldest boy, he was like 2, and I taught him how to multiply, and I don't think you could add.
It's just like procedural things, right?
Yeah.
For the most part. I don't know.
Yeah, so,
Man,
you know,
I would-
do we really have to teach these things the way that, you know, bore so many students?
I mean, what's unfortunate is we're teaching to the average, right?
And maybe you are. Actually, average is pretty good. I
Yeah.
think average does pretty well. And, you know, and I don't think we need to say, well, we have to fail X number.
Yeah.
You know, that I don't know any faculty member where that's in their scheme of things, where there can only be so many A's and there has to be so many F's.
Yeah.
You know, you know, I always try to impress upon my students that, hey, if everybody gets an A, then either my exams are too easy or we're all doing great jobs. You know, so
It's hard to figure out which one of those it is. But no, I agree.
it hasn't happened yet, but one section of one class that I've taught for years, I had 2 to 2.5 times more A's in that section than I ever have.
How many students?
There were about 60.
Okay.
And so it wasn't a giant section,
Yeah.
but it was a decent sized section.
I was going to say, if N gets over 200, I think it's your population pretty consistent. I mean, at least in terms of my
Yeah.
numbers, in terms of A's and what.
Well, I get to do 220 again this semester, so we'll see how it matches
Yeah.
the other times I've taught. But I'm not sure there's a big difference between 165 and 220.
What are the courses? What are those? Students?
Genetics. So general genetics, required of biology majors,
I see.
and there's lots of bio majors, but students going to medical school, biochemistry majors take it, computational biology majors, neuroscience majors, exercise science. So lots of students in the life sciences take genetics.
All right. Contra has a question. Can
Yeah.
nanoscale lipids enter bacteria?
Enter. Bacteria are really good at keeping all life is really good at keeping things out. So just plain old naked lipids, but bacteria will take up. So they have mechanisms to actually take up really tiny lipids from the solution around them. And whether they're actually slipping through their cell wall, there is evidence for uptake, and then we can trick them so that it probably a combined process of fusion and uptake. That's how we get foreign DNA into bacteria.
So uptake in this case means through the membrane.
Through the membrane in an active biology
I
process.
see. Not just like absorbed
Not
on
just
the surface or something.
Right,
Yeah.
right. Yeah, it's hard to get lipid membranes to absorb, right? Because they're two greasy balls. And so they don't necessarily want to go together because that water shield between them, thermodynamically, especially on very small droplets, is an impressive amount of energy needed to get those lipids to fuse.
Yeah,
I don't know any more physical chemistry than that.
But one of the big breakthroughs with the RNA vaccines was the transport, right? It was the
yeah, so it was using lipids these same lipids. So, you know, we mixed DNA with lipids to get them in bacteria. I don't know how to do this one.
I think it's just follow the arrows when they pop up, but they're it's not messing around.
Yeah.
I'm curious if it's the same sequence or not. Don't know if that's the case.
Then we use lipids to get DNA into single cells, just cells in culture, so that we can get cells to carry, and then, but naked RNA causes an immune response, and the RNA falls apart really fast before it gets into cells. So it itself is a bit antigenic, and so having it I'm going to get killed here because I don't know what I'm doing.
No worries. Keep in mind, this is Jim's first time seeing this game. We're just playing this one for fun because it's just a fun shooter.
You know, it's a game. Yeah, but with the viruses, the RNA viruses, that lipid, you know, it binds to the lipid, it masks the RNA so that our own immune system doesn't break it down, or enzymes don't break it down. You know, we don't want free RNA in us. So we have lots of RNases to break down RNA, like on our fingertips, etc., because RNA viruses tend to not be healthy for us. And so they had to shield that RNA with these lipids, and then that also helps it get into the cells that are local.
Does this one actually control steering? It might.
Yeah, I think this one controls steering.
Oh, yeah.
Otherwise, you crash into trees.
Yeah. So this is the only one where you actually get to steer. The rest of them, it's just on the rail. Huh. Contra. This isn't how you beat Saber. That's awesome. Beat Saber is I don't know if you've seen this. The Oculus virtual reality, where you have like swords and you're hitting things that come at you. That's kind of what that. Yeah, that's just not Beat Saber for sure.
Okay, yeah, that was not an enjoyable game. Okay, and these trees come at you too fast. Now I feel like this is why I don't downhill ski. Lived in Utah and don't downhill ski because there's trees.
Oh man.
And again, I do things at work that are slow, so I cross country ski. Or slower yet, you snowshoe.
That's fun.
Yeah, cross country skiing's fun. You know, you can get places where you can't get walking.
Yeah, and it's just a super relaxing.
And it can be a workout. If you decide to go, then it's a great workout.
Yeah, a lot of people, I mean, if you don't grow up in snow country, don't appreciate how just like quiet and calm it is after the big snow. It's such a unique.
Yeah,
you don't hear any bugs. You don't hear
I'll give a plug to the national parks, because when you're not at working, then you find extra time to do other things, like go to national parks, and there's lots of them out west. So we went to Yellowstone in the winter. It's open seasonally in the winter. And there's more visitors in a day to Yellowstone than there are in the entire winter season. Then they shut down the park to get it ready for spring and summer. Then they shut it down again to get it ready for wintertime. So so that was fun.
That's awesome.
Going there, and we were on a trail that nobody had been on for several snows. And so, you know, using your little Pathfinder on app, All Trails or one of those, so that you can stay at least close to where the trail is while you're out there. So you just hope the person that you're following actually stayed close to the trail.
There's a lot of trust in that.
Somebody who followed, and you hope that you're following the one that finished the trail and just didn't go off on their own tangent sometimes. So, but it was, that's.
All right, I'm going to run to the restroom. What do I give you for a question before we go? Favorite equation. Do you have a favorite?
Oh, I don't know if I have a favorite equation.
Maybe not that one.
Let's see. Favorite equation. Can I think about my favorite equation?
Yeah.
What equation I've been using Pythagorean theorem.
Interesting.
Recently, for reasons. Yeah, if somebody can guess why I was using it, then that would be remarkable.
This is related to your research?
Define research.
Research loosely defined.
I was purchasing a gift. So I needed to use the Pythagorean theorem.
Huh. Now I need to know the answer. What is it?
I was trying to maximize the surface area of a steel pizza pan versus a circular pizza pan versus an oval pizza pan. So I had to use, elementary math.
Yeah. A lot of geometry.
Yep.
That's amazing.
So it was really fun. Now, was this work? I was doing it at my desk on sabbatical because I remembered I had to send a gift.
Technically, you got paid to do it.
And, well, I don't put it that way. I was on sabbatical.
Yeah.
And I ended up not buying a pizza pan. I bought a stainless steel or a steel skillet because I thought that was an easier one to size.
That's fun.
Well, if you're buying a pizza pan, is it going to be on the grill? Is it going to be in the oven? What size is the oven? Some fancy ovens actually aren't as deep as old school gas ovens.
Oh.
And if it doesn't have a handle, how are you going to get it off your grill?
Yeah.
Because these are not.
No, these are
light.
practical questions.
These were very important questions.
And we need a Pythagorean theorem.
So you have to use the Pythagorean theorem.
Oh man, no, you got Darth Vader.
Yeah.
So this is going to be similar to the. Oh, you blocked it. Nice.
That was luck.
We'll take luck.
Yeah. Oh wait. Okay. Forward is down. Look at that.
Oh, it's.
I feel like I'm on the spaceship in. Yeah, forward's down on an airplane, but in Independence Day, it's reverse.
Yeah. That's we all learned from alien technology.
That's right.
All right.
That's how you know if you're in an alien spaceship.
Oh, let's do this one while I run to the bathroom. What's the Earth is flat or anti-vax equivalent in your field? Could be anti-vax. Well, what dogmas exist? What would you like to dispel?
Oh my gosh. And he leaves me. Oh my gosh. So now I'm left here with Darth hacking at me because I'm sliding the mouse around is not the same as working with the joystick's too jumpy. So I can't use the joystick. Well, maybe the joystick is better. I don't think it matters if I'm using the joystick. So what would I like to dispel in the field of biology? Oh my gosh, there's so many things to choose from. Somebody give me an answer for what should be dispelled in the field of biology. I don't think of these terms. I'm not like a hypothesis-driven scientist. I do what we call fishing expeditions. No pun intended. We set up systems, and then we look to see how those systems are altered, and then we go after these events that occur for lots of our research. So we mutagenize randomly, things like that. And we look at the same system again and again. I don't think I have control enough to switch out of this game. Oh, there we go. So I asked for help online, but I can't read your screen over there.
No worries.
So they get to answer what would they like to see as proven in biology.
I have to do this comment first. Nobody told Luke the lightsaber was y-axis inverted, and that's why he lost a hand. Confirmed lore, thank you, Contra. That is amazing. I mean, there's a lot of public misconceptions about genetics. I mean, everything from, like, autism being caused by vaccines to...
Yeah.
Like, I mean, you could pick any one of those.
I mean, even... one of the areas that I touch upon, not heavily, because it's one of those where you have to get a little bit more genetics under your belt. But even the notion of like race, ethnicity, and there's more variation across breeds of dogs than there are across all what we call races or ethnicities in humans. Humans are so much alike, no matter what continent we were plucked from. Because there's always been lots of admixing, and we have really slow generation times. And so it takes a long time for genes to get fixed in a population. And then once they're there, it takes a long time for them to be removed from a population. So, it's all the same things happen: mutation, drift, migration, as, other species. Yeah. It's just, we have lots of other things going on, societal constraints. We're not limited by geography as much as most species.
Yeah.
And, but yeah, humans are really humans. I mean, we are a species where we don't vary a lot genetically. It's just that as humans, we're really good at picking out those features where we look different.
Yeah.
Right. But even that isn't always the same genetic basis for like pigmentation, things like that. There's lots of variation. Those things vary. See, the things that we see normally vary, but the rest of us doesn't vary a whole heck of a lot. We're just good at finding those subtle little differences. But dogs vary more than humans, and humans have been around longer than like all Canis familiaris or our dog breeds, because we've selected for those breeds.
Yeah, I was going to say, you can do that, but it has to be very targeted and very restricted.
It's very targeted, and you're basically eliminating those things that you don't want, and you're favoring those things that you do want. And then there's lots of notions like, oh my gosh, what dogs have these certain traits because we selected for them. Then, there's stories and papers published from 100 years ago of recouping modern dog from fox or this, and certain traits are linked, but they turn out genetically they're not linked. And so those may have just been, in those small outcomes, those experiments, they happened to be in that probability zone where, it looks like this is happening, and it was. But if you did that experiment a bunch of times, that would only be the result of a small percentage.
So there's a classic one out of Russia, the tame fox.
Yeah, the tame fox. Yeah.
So is that a real one? Is that a?
Yeah, those traits aren't really linked the way that we thought they were from the tame fox.
Interesting.
Yeah. So wagging tail, smiley faces, ears back, and all that.
So anyone that's not familiar, I mean, can you give context into this, because it's a very famous one, right?
Yeah. So it was one of these where they tried to recoup and try to understand the origin of the modern dog, right? So how did humans select for these traits? And most dogs came from wolves. But there's lots of admixing, especially early on. So the dog geneticists, which is a really well worked out field.
I was going to say, they have a good data set.
They have great data sets. We've done lots of inbreeding, so these poor species are bottlenecked into having lots of diseases, 80 or 90 genetic diseases that lots of humans have. So they're a useful model for studying disease. Primarily because we pigeonholed them in these odd ways. I don't want to shoot them; those are my guys. And but so could they recoup traits similar in the dog from Arctic fox? And the story was, they got a lot of things out of the Arctic fox with selecting these traits. But those experiments are really hard to reproduce. One, they were done a long time ago, so the standards for how we did experiments and how we actually did our statistics, randomized groups, we didn't do that back at those days as scientists. We've learned that it alone causes lots of variation, so we really want our data to be reproducible, our experiments to be reproducible. And when things aren't reproducible, it's not because somebody was usually fabricating data. It's that experiments are hard. And there's lots of things that we don't know we're controlling for in my lab versus somebody else's lab.
I was going to say, and the standards and procedures, they just didn't exist, right?
That's right.
Wild west of experimentation.
That's right. Which was great because it led to lots of interesting outcomes. That when we look at it, more refined aspects, then there's a lot more subtlety in these, or they were doing experiments that there was an inherent bias that they just didn't know existed.
So sorry to interrupt, Jim, but Actual Education rating with a party of 12. Thank you for joining us, Actual Education, a fellow educational streamer. You guys should check them out. They do math and science tutoring on Twitch, which is kind of fun using this as a medium. The kids really like it, so you might as well. So Actual Education, thank you for the raid. Our guest today is Dr. Jim Fadool. He's an expert in neurobiology, and particularly zebrafish and zebrafish genetics to understand vision, retinal generation, degeneration as a model for human behavior, and hopefully doing something useful for humans, but also learning a whole lot along the way. If you guys have questions, feel free to throw them in chat. Nuclear demonstrations, thank you for the follow. Thank you for joining us. It's awesome to have you here while we play a little bit of Star Wars and we bring down the Death Star. So it's right click when we have it come up. We're going to do it this time. We're going to save. We're going to save it. I'm going to wait for the Millennium Falcon. But yeah, actual education. I hope your stream went well. The fellow Wednesday night streamer. It's an unusual night. This is our last week of freedom before classes start at FSU next week. So yeah, we're going to celebrate, enjoy, take some drinks, relax, have some fun. Let me catch up on some. The placebo is strong with this one. That's awesome.
Get in the wall.
Oh, you got it?
Got it.
Holy crap.
I just saved millions of lives.
Exactly. I did not hear them scream. They all celebrated. It's amazing.
They didn't know what was going to happen.
There's a recent study on fish being able to feel pleasure. Are you familiar with this?
I am.
Wow, that's awesome.
I am. So I haven't read the full paper yet.
But before you go on that, easy as a womp rat, absolutely. Congratulations on nailing the 2 meter wide hole. Good game, Alderon. Oh, it's the little victories. But anyway, sorry, we got fish being able to feel pleasure.
Yeah. So
which fish?
So it's a I didn't read it that carefully. I would have to pull up the paper. This is why we have the internet, so I don't have to remember all these details.
But broad strokes for those
broad strokes. So it's a fish that uses a cleaner fish, and how does the fish know where to go to get cleaned? How does it the fish that's going to go to these cleaner fish? Well, it turns out that the act of getting cleaned, so the cleaner fish. Then I think in the paper they say massage, or in the blurb I read they say they massage the other fish, and it causes stimulation of the same neural circuits that cause kind of pleasure for us or opioid addiction in us. So it's these dopamine circuits. So you get this rush, and so these fish, after one cleaning in a tank, then they go back to the same place to get another cleaning. And then if they add opioids to these fish, then it stimulates that behavior. And if they block it with Narcan, then the fish don't seek out that behavior.
They don't detox them.
They can detox them. Now, is it pleasure seeking? Well, do fish feel pleasure? I'm not sure.
Interesting.
That's beyond my understanding of fish.
I mean, that's our anthropomorphism of them.
That's right. That's right. But I think the more important thing is this use of this neurosystem is ingrained in vertebrates. So it's nothing unique to humans. It evolved likely for some purpose that was useful and it stuck around. So cleaning fish is a very important aspect of fish life for many fishes, right? Get parasites off their skin, fungus off their scales, or from in between their scales. And so this would tie something that's needed to a brain circuit that triggers a certain behavior. And then that's been used in us. And, we do have opioids and keflins in our own systems. And then when we add exogenous opioids, then, we're hijacking that system for a different purpose. So I think it's interesting because this isn't just something we find in mammals, but even in fishes. But the lab that I trained in Boston, there was a postdoc that joined the lab just after I left. And he was interested in addiction. And they had money from one of the National Institutes of Health, basically to test if fish could be or demonstrated addiction. And so he set up these behavioral chambers, and it's very hard to do these types of behaviors because, fish are always on the lookout for something that's going to eat them. And so when these experiments were done, it essentially, and this fits these data, that it just took one exposure of the fish to, in this case, it was cocaine, and the fish would return to that side of the tank. So it's a little bit more elaborate paradigm.
Yeah.
So, it's a T maze, and one side of the T has marbles that are blue, and the other side has triangles that are red, something like that. And one side it has cocaine and one side doesn't. And the fish would then seek out the objects that were in the cocaine side after one exposure to cocaine. Right? So this fits the data that we knew from 20 some years ago, or actually, 25 years ago, almost 30 years ago, that fish displayed this ability for it's called place preference. So they will return to a place where they were exposed to this drug, and so if you want to say that was a demonstration of pleasure, I don't know if that's accurate. Right? Perception, that's us putting our emotions on a fish.
Actual education followed up with 2 important questions. Should we stop eating sushi? Jim Fadool, on behalf of all biologists everywhere.
Why should we stop eating sushi?
I mean, if they feel pleasure, do they feel suffering? I mean, this is a bigger question. I've had this quite a lot at actually faculty dinners, because we all have this spectrum of what we're willing to eat, and often there's a line drawn on cognitive awareness, right? Where we won't eat human, primate, dolphin, dog. But where do you draw that line? And for me, I won't eat octopus because they are
octopi.
Very smart.
That's that's been recently added to the list for many people, and even squid. Now another cephalopod. They're from the same larger clade. So it's
not necessarily that they're going to feel me eating them, but like the intrinsic need to farm them, and you're essentially keeping this cognitive creature in a cage.
Right.
Their entire lives is where I draw
or just
the line.
going out to harvest.
Yeah.
Even in the case of octopi, and so a lot of people don't eat lobster because I worked in a restaurant in New England, so I prepared lots of lobsters, and and then our sensibilities shift.
Yeah.
For many people,
So
in both directions.
So do you mind sharing where your line is on that spectrum of
Eating.
Yeah. What's acceptable to eat? What's the highest cognitive? I mean, fish I don't feel bad about.
Yeah, so I eat. Yeah, I eat just about everything. I don't eat primates.
Yeah.
But I don't eat elephant. Never been offered elephant. Actually, there was a bar in Detroit that had a can of elephant, and you could order an elephant burger.
That'd be a hard one.
Yes, and that had to be a very old can because it may be against the law to still have that can of elephant.
It's aged to perfection.
And but no, so it makes me think about it. So since with the octopi, so I haven't had anything octopus in a very long time. But see, there's so many other things that I enjoy eating, so I don't have to go out of my way. I never wake up in the morning and say, I have to have octopus today.
Yeah, I was gonna say it's.
Do I really need a squid today?
Of the appetizers you could choose.
Of the appetizers on the menu, and I'm not one of those that says, well, it's already been brought to the restaurant, so somebody's gonna eat it.
That's a rationale. All right, it is 10:30. It is time to transition over. Are you ready, Jim? We're going to go back to the Nintendo controller, the crippler. It is time. We will revisit your questions, but on Ask a Scientist Gaming, we have a tradition. We close every single stream with one particular game, and that game is NARC for the Nintendo Entertainment System. So I'm going to get this set up so you guys can see it.
The last time I played NARC was sitting in this chair
4 years
in front
ago.
of the green screen.
Yeah, probably last time you touched that controller was 4 years ago.
Actually, you loaned me a controller so I could practice. So I have one of these in my office.
That's awesome. And once in a while,
I pull it out once in a while to do something with it.
Before you press start, everyone, this is NARC. This is the tradition. Jim is going to play this game. Not only is he going to play this game, but he's going to race against his personal best, but also his colleagues across all disciplines, mostly at Florida State University, but also across the country. So we're going to put up a clock. I'm going to get that on the screen right now. Jim, do you want to see the clock or not see the clock?
It does not matter.
No,
it does not
no
matter.
strong preference one way or the other?
None.
All right, we got
It's
the
just
clock.
like walking in a restaurant.
All right, so I'm going to put the clock over here, and I'm going to read for you guys. Here's the Nintendo cartridge, the original box art. You can see it right there. NARC, no one had the guts until now. Jim, you have the guts. And so you are Max Force. Your mission, bust Mr. Big and destroy his dreaded criminal empire. Seize all contraband, stolen money, illegal weapons, use rocket bombs, high-powered machine guns, apprehend all suspects, protect the innocent, punish the guilty, stop at nothing. So go ahead and press start whenever you're ready.
Explain to me what I'm pressing.
So this button right here to get going.
Yep.
And then B.
The one that says start starts the game.
Start is the game. And then B is going to be jump. If you tap it, it's jump. If you hold it, it's squat. And then A is if you tap it, is a rocket. If you hold it, it's bullets. And so the tapping is what you're going to have to get used to. So it doesn't matter. Mostly walk right and keep going, and you will
mostly walk right.
That is about the extent of the game. There's only one part you need actual engagement with the game, and we'll get to that as we do. So go ahead and press start, and I will start the clock as soon as you hit the ground running. We are going to win the war on drugs, ladies and gentlemen. It is coming. All right, there we go. The clock has started. And if you guys have any questions for Jim and want to distract him from his really important endeavor to get the best time, go right. No advantage in killing anyone. Just walk right, find a door. Your taps are good. So yeah, the big thing you're going to have to do at the end is tap jump and then rocket. And so you're going to tap B and then tap A.
Oh, and I have to get to that door.
Yep, so walk through that door. Then you'll go right. You'll find a card on the ground, and you will keep going, and you're going to do that several times in a row. All right, I got to catch up on some things too here. Nuclear demonstrations. We do not have the subjective experience of fish. This kind of thing summarizes interpretation of research in general. Agree. This kind of thing summarizes interpretations of research in general. Agree. Interesting. So you're going to hit, hit that start button again. Get that guy right there.
That guy?
The card, right? Pick that up.
How do I pick that up?
Over it. Yeah, there you go.
There we
All
go.
right, find a door on the right, and then keep walking right. And here I would eat dog, hypothetically. Eating primates is just a bad idea from a safety perspective. Prions, yes. that's a fair critique.
And that's another reason, yeah. Right.
All right, Actual Education had a question a while back. Who would win, meth bear or cocaine shark?
Are they in the water or on the land?
I was going to say, it depends on the venue, right? Like, it's in the jungle.
It's in the jungle. I'm going to say shark.
Meth bear, cocaine shark. Actual Education coming in asking the important questions. Nuclear, thank you for the follow. Thank you for joining us. Always a pleasure to have a fellow education raid. Lori has requested a factoid. Jim, do you have a knowledge bomb you'd like to drop on the audience?
Oh, I wasn't prepared for this.
Oh, no, I didn't give you the reminder, did I?
You may have. I don't read email.
Yeah, that's fair. And these emails are quite lengthy. Fortunately, you get the copy and paste. But do you have any interesting factoids, things you learned? Actually, we could turn one of these into it. Maybe that one?
Oh, yeah.
We don't have to ask the question.
This was one of my favorite things. So in animals or plants, there's what are called chimeras. So if you think back to mythology, this was this mythical animal that had
you're going to shoot these guys wearing black with bullets at close range.
Okay.
And one of them's going to drop a card. So bullets, there. First shot, get it. Blue card, and then through the door. Hit that slot. And through the door. That was quick. Nice. Speed strats. All right, sorry, chimeras.
Chimeras, yeah. So there's chimeras in populations, in the human population. So these are single individuals, but they're made up of cells from more than one individual. So this would be, there's these notions that everybody had a twin, and then all of us absorbed a twin, except those that didn't absorb their twin. And so the chimeras in the human population, and we thought it was pretty rare. It turns out there's micro chimeras. And so during fetal development, cells from the fetus pass through the bloodstream to mom. So all women
jump in that car, and then tap B.
B. There you go.
Stay on the bottom and go right.
And so all women who have had or been pregnant, then they carry cells from their child with them.
Oh, interesting.
And so they're called microchimeras, and we can only can detect these now because of all. I want to jump over those.
But that's an outcome of pregnancy, then.
That's an outcome of pregnancy, and so there's some evidence that even the fetus is then carrying some mom cells.
Huh.
So then maybe all of us are microchimeras.
That's interesting. So micro implying there's some portion. Presumably there's macro chimeras as well.
Macro, yeah. There's several famous instances where one, I can't remember the woman's name, but her husband filed for divorce while she was pregnant, and then when the baby was born, he claimed that he wasn't the father, so a paternity test showed that he was indeed the father, but she was not the mother. And so her children were taken away from her. She was not a woman of much financial means, and then she was pregnant again, and so the court assigned a person to watch the birth, stay with the baby the whole time till it could be tested, and it turned out that the baby, this baby was her baby, came out of the woman, but it did not have her DNA.
Oof. Jeez. How long ago was this?
The 70s.
Wow.
And it turned out, so she was a chimera. She must have had a fraternal twin that was absorbed.
Nuclear said they charged her with fraud.
Yep.
Wow.
Yeah.
Holy shit.
Now, similarly, there was another woman who had, she needed a transplant.
Tap B. Stay on the bottom. Oh, a little lower. There you go. No, you were right. Press down a little bit. There you go. Right there. Whoa. All right.
There we
Stay
go.
on the bottom. Go right. Go up to the top. It made it onto an episode of House. I do not remember it. Oh, no, where's the wall was right up here. You're not going to win the car battle, unfortunately.
No.
Fun. Wow.
Well, this other woman, she needed either a transplant or a transfusion, and they looked at her children, and they were a different tissue type, so it didn't match.
Oh.
And it was the same father, and it turned out that they had a biopsy sample from before, and the biopsy sample was the same tissue type as the children, but the biopsy sample that they used for the tissue typing was either skin or blood, so it was a different.
So get in the car, stay on the bottom, and you'll just ride it out all the way to the end.
Yeah.
All right. And Lori redeemed Take a Drink, so cheers, Jim. Hopefully you're having fun.
I am.
Visiting some retro games. Man, that is crazy. Nuclear had a follow-up on that. Isn't it hard to detect in general? Lots of testing is needed. I guess, how do you detect that? How do you detect two independent chunks of DNA?
Yeah, so it's only fallen out in cases where there's been reason that somebody's going through tissue typing, paternity or paternity tests, not maternity, because we usually know who mom is. And what are the other cases? But now with genome sequencing, not everything fits like family stories.
Interesting.
It's yeah, so I had a friend who he had a PhD and he went to teach at an exclusive prep school in New England, and they did fraternity. They did, the blood typing. You do it by DNA, just to teach.
So go up through the door, but don't press two up.
Oh,
yep, that happens. So go through the door, but don't press too far up. Further to the right.
Oh,
last door on the right.
Oh.
There were a lot more people claiming to be descended from royalty before the test, for sure.
Yeah.
Just go left, you're gonna hit that. Man, that's interesting. So the chimera prevalence, I mean, you had an actual number here. It's greater than 5%. Or at least that's the general hypothesis that encompasses.
Yeah.
Leslie Magenta, thank you for the follow. Thank you for joining us. We're in the last minutes of Ask a Scientist Gaming, Mediocre Gameplay, Expert Science. We got about 15 minutes left in the stream. While Jim plays one of our classics, NARC, we make every guest play this at the end of the night, and we have a brace. But in the meantime, if you want to mess with Jim's gameplay, just ask him some questions, whether it be about food or alcohol or neurobiology. He's happy to answer those. He's an expert in neurobiology, particularly zebrafish use those as model systems. Sorry, yeah, this one you're going to go to the left. Yeah. And you're going to shoot the green guys with bullets at close range. So then shoot the white guy and then turn around. Then bullets when the green Rambo guy comes out. These are all great questions tonight. Contra, thank you for the comment. Thank you all for the questions. So yeah, shoot. Oh. So you're gonna shoot him with bullets and then the Rambo looking character. There. All right, green card. You've had really good luck with cards tonight, so congratulations on that. GG's.
I think that hung me up last time.
Yeah. No, it's what we call in speedrunning is RNG, random number generated. You have a 1 in 6 chance of that and a 1 in 8 chance of the blue card. And I think you got them both first try. So shout out to you on that one. All right. Yeah, Leslie Magenta, thank you for joining. First time chat. Always need to distract speedrunners, especially. Oh man, I got some speedrunning news for you guys. Let me bring it up here. So we had a guest, Chris Martin. He played NHL 94, and the reason I knew about NHL 94 is because I played NHLPA 93, and so there was a trick in the game that I learned about when I was young. That when you use so it's a hockey game, right? Yeah. And so if you use the player 2 controller, even if it's a player 1 game, you can use the player 2 controller at the start of the game to remove the goalie of the other team. And the goalie stays out the entire time. And so when Chris was playing NHL 94, I tested it on that, and I tested it on NHL 93. But then I also learned there's a speedrunning category for how fast you score goals in the game. And it turns out the previous speedrunners didn't use this trick. Sorry, go right. There's a door on the end. That guy's the door. There it is. Yep. It's so dumb, but that's true. But what I did was I used that goalie pulling technique, and I timed how fast I scored goals. And now, ladies and gentlemen, I am the world record holder in the NHLPA Hockey 93, 10 goals and 5 goals scored. As of 2 days ago, they verified the run. And so, better living through cheating. I don't know how we describe it, but yeah, this is why I love speedrunning. I'm not good at the games. I just figure out exploits that make me faster than other people. So
better living through electrons.
Exactly. So yeah, now I'm the world record holder in those categories. It won't last long because the people I beat will figure out this trick and use it, and they'll do it better than me. But nonetheless, I count as a world record holder, at least momentarily. So shout out to speedrunners there.
You could have put that in your tenure binder.
Should have. See, it's going to go in my retention request. Like, do you really want to lose a world record speedrunner?
That's right. I don't have to do that for 5 years though.
Now, applications of a new glitch technique is 100% legit. Exactly, and that's the thing. When I looked at it, I was like, either they have to change the rules or they have to give me the time, and they gave it to me. So yeah, we'll count that as an exploit, and that's completely legit.
So now can they exclude other people from using that exploit?
No, if they do, then they have to take away my time. So you have the blue card, go ahead and hit that. But yeah, sorry for the distraction. NHLPA, obviously near and dear to my heart, but we're going to answer some questions. Contra had a question a while back. Do those things found on fish, funguses and parasites, differ from those in the wild versus captivity? Ooh, I have to assume yes, right? It's all environmental.
But I think what we have in captivity
came from the environment.
They came from the wild.
Yeah.
Yeah, so I don't think we've had fish long enough that we now have captive. There may be strains that are only found in fish species, but most our pet store fish are coming from giant ponds in like Florida. So, these are typical of environmental strains that then invade our fish tanks.
All right, so now we are on to Mr. Big. So yellow. Kill these guys on the screen. So yeah, stay in this area, shoot everyone on screen, and what's going to show up is Mr. Big in a wheelchair, and you're going to have to shoot him with a rocket. So he's going to come flying in, so tap rockets a whole bunch. So gentler, yeah, there you go. Yes, ladies and gentlemen, every other Wednesday night we shoot a guy in a wheelchair with rockets, and it just works out. I recommend standing like up here. It increases the odds. So kill everyone on screen. He'll come back. Nuclear has a good point regarding the speed running. When you find a new glitch, if it's a popular enough game, it'll be a different category. They'll like split it up into like glitchless versus glitched. But this game isn't popular enough. There's only like 5 people on the list. But guess who's number 1, ladies and gentlemen?
So are those true glitches, or are they?
I mean, I don't know.
Or are they cheats that were written in? You know, if you ever apply for a job with the CIA, you look at the code.
Yeah, I don't know if that was intentional, like pulling the goalie. What's really interesting about it is if you do like playoffs, right? If you do a tournament, early games, if you pull the goalie stays out. But later, as the team gets better AI or whatever it is, or better, responsiveness, they put the goalie back in. So I don't think it was intentional. Otherwise, I don't think that would exist.
Yeah.
But I don't know. But yeah. We'll count as an exploit. So nonetheless,
We'll pretend like it was intended.
Yeah, exactly. And I'm one of the few to use it. There we go. Now he's going to inchworm off. We got to do that 3 times total, and then we'll go on to the next boss. But yeah, if you guys have any questions for Jim, one of the ones I'm going to ask, particularly in the last 10 minutes, which is one of my favorites. I honestly don't remember what we came up with last time, but I love movies. I love science captured in movies, well or poorly, because it's entertaining either way. So for you, when you see a movie, there's certain ones you saw were like on point, related to your expertise or discipline, and other ones that are completely off. So what movie or TV show got your discipline right? Which ones got it wrong?
It's a question.
Yes.
Okay, Jurassic Park.
Yeah.
Got right that we can extract DNA from these amber embedded
rockets
animals. Can you go out and build a dinosaur based upon a frog genome? I don't think we can do that.
Not yet or not ever?
There's this company that supposedly made a wolf? The wolf that's in
the dire wolf.
Dire wolf, yeah. It's not a dire wolf. It's a wolf that expresses a handful of dire wolf genes. Right? So, and they picked the ones that they targeted, those that would make it look more.
There you go. Stay left. He's going to drop a card, and if you go off screen, you'll lose the card.
There it is.
Right there. All right. Grab the card, go right. We're on to Mr. Big, Phase 2, ladies and gentlemen. So Jurassic Park got some things right, but got the central theme wrong.
But, it was pretty cool.
Yeah. Any other ones you can think of?
Oh, The Fly? The original Fly.
Yeah.
So I was fascinated with that movie as a child.
So you're going to go to the top of the screen, and you're going to jump rocket him in the hat. So you have to tap B and then tap A. So yeah, maybe just run this way for a while.
Okay.
Get some separation. And then you can turn around and just stop walking.
And what do I do?
So run away. Get some distance.
Run away.
Because if he's on you, you just can't do anything. You see
right.
So get some distance. Even if he's off screen, it's fine. You got a lot of distance to walk. And just stop walking, but turn around, and then tap B, and then on the way down, tap A. So you got to rocket him in the hat. And this is the rate-limiting step in this game. If anyone's played this is so challenging to do because it's just such an unusual move. But you're going to walk away, jump up, jump, so tap B and then tap A. One way to do this, actually, is to put the controller down and, like, piano it. Because you're going to, like, it's tap B, and it's a very gentle hit. Texan Space Agency. Hello, friends. Hope you and yours have been well. It's good to see you. It has been a while. Thank you for joining us. Thanks for joining us, Dr. Fadool. Absolutely, Texan. You're in here the last 10 minutes, but if you have some neurobiology questions, Jim is happy to answer them. We're talking about movies right now, particularly Jurassic Park. How it got some things right, got some things wrong. And then we talked about The Fly. Why is that one of your...
Yeah, so in the original Fly, same thing. Fly gets in the transformer machine, but it turns out that it's like the fly parts are big, and then at the very end there's a tiny little fly, but it's a human trapped in a spider web, and it's saying help me. And But so you had you had a chimera, right? You had a fly that had the giant fly was the man, but he had human parts, but it was big size, or you had the, whoops. Yeah, this is.
No worries. If you get bored with it, I can intervene and get you through this stage of it, because it is a nightmare. So that's the 1958 The Fly, right? Because that one's the one that ends in the...
That's right. Where the modern fly, the Jeff Goldblum fly, then they're merged, and then he's slowly turning into a fly as fly genes are expressed.
Which one's more realistic?
Well, it all depends on the transporter machine.
If you had to choose.
It depends on how because, we have a lot more cells than a fly.
Yeah.
So, I mean...
I was going to say there's mass balance issues.
Yeah,
though, right?
That's the problem. And then, you turn into a baby human with fly wings and some fly legs.
So unrealistic.
But it's an interesting concept when I was, it came out in 58.
Yeah.
So I was -6, -5.
Solid.
So probably watched it when I was 9 or 10.
Formative years.
It was cool.
Yeah.
I mean,
that's a career in genetics. That's awesome.
Yeah.
We're going to say it's causal. We're going to say this is the origin of your interest.
So I'm still on the way
close.
I'm still on my way to making the perfect chimera. But we do this in fish. We can do this intentionally. We can merge 2 embryos or do cell transplants.
I mean, the slow expression of the traits in the new one is intriguing, right?
That is. So
I mean, is that something you could splice in and just weird stuff happens over time?
I'm not sure. There's an experiment that's really cool, and it's there's a gene in us called PAX6. When it's mutated, then those individuals have what's called aniridia, so it's a missing iris or a part of a missing iris, so it's involved in eye development as well as other organs. And then in the mouse, that same gene, when it's mutated, it's the small eye mutant mouse. And in Drosophila, when it's mutated, then it's called eyeless. And there was a great study that was done where they substituted the mouse gene into the fly. And the mouse the fly with the mouse gene doesn't make a mouse eye, but that same gene turns on the fly eye program, so you recover fly eye. So that's why expressing fly genes in us, if they were similar enough, it might turn on our gene that was the target. But would you really get a whole fly?
Only one way to find out.
So it's interesting. I think it's an interesting concept because we can do it gene by gene when they control. The fly eye doesn't look like our eye.
Yeah.
But at the top of that genetic program, it's the evolutionarily the same gene.
That's really intriguing.
But it activates fly eye, right? Because the fly doesn't carry the rest of the mouse eye genes. But in this case, we've put in the fly eye gene. So could you really make a fly eye?
So sci-fi preempted this idea.
Maybe. Then there's that darn immune system.
We'll ignore that.
Yeah, so let's ignore immunology. I didn't take immunology, so we can ignore.
AJ Brooks, thank you for the follow. Thank you for joining us in the last minutes of winning the war on drugs.
How are you doing? Oh
Dr.
my gosh.
Jim Fadool is working against Mr. Big. This is the hardest part. It's pretty terrible.
I keep jumping into Mr. Big.
Man, we had somebody beat it in 12 minutes because they got this first try. That's it.
It was a glitch.
I volunteer for Fly Eye human trials. Thank you, Texan.
Okay, I'm not going to write the protocol to get that approved.
Yeah, where's the IRB on that one?
Oh, you need both.
I was going to say, you probably need an island somewhere that's not restricted. Do this research on them both, but that's awesome. So, the Fly, kind of good, at least intriguing in terms of the use of genetics and genetic engineering.
It's a very interesting eye on the fly.
AJ Brooks, you and my homie would totally get along. He's got a near complete NES game collection.
How did I miss
that? Man, that's really fun. So I just crossed 400 Nintendo games in our collection. And so one of my favorite hobbies is anytime I go to like a new city for a conference or give a seminar or whatever it is, I find a retro game store. And so I always hit those up and try to find, and I have my little spreadsheet on my cell phone and try to find new missing games. But yeah, that's
Do you
pretty
play
fun.
the games?
I play them at least once in the cartridge form, but I have emulators that do all of them now. So it's like I can play them here much easier, less blowing on cartridges. Man, that's pretty cool. Missing 2 to 4 games. There's some Nintendo games that are like thousands of dollars now. They were so rare that, yeah, I don't think I'll be collecting those.
We should look in our pool.
Yeah. Man, garage sales used to have a lot, but that's gone out. Last time we hung out, he was playing Pipe Dream. I do have Pipe Dream. I have that on the shelf. Yeah, you'll see here. The NARC cartridge, because this is our favorite game. And I'm the former world record holder in this one. No longer the record holder. I got beat at this.
This is taking a long time.
Yeah, you want me to intervene?
Yes.
You don't have to. So it's annoyingly specific, but it's that shot right there. All right, I'll get you away from him so you can do some bullets to the skeleton. All right.
I was shooting too soon.
Yeah, the timing on that and the
Yeah.
tap is pretty terrible. All right, so I press pause. So press start. You're going to shoot bullets at him in his vertebrae and move it up and down, and you can. So press start and then just hold the A button. Run away from him. There, you got one of them. So you just have to do that while moving up and down a little bit. Texan Space Agency. Emulators are cheating at work. We can have an internet connected device, so I game on a Game Boy Color. Emulators are not cheating, sir. Well, it depends on the emulator. Turns out some of them are exact recreations of the hardware in silico, which is pretty amazing. But yeah, Game Boy Color is pretty fun. But yeah, just run away. Turn around. It helps to be in between these two black lines for unknown reasons to me. There you go, another vertebrae gone. All right, last minute questions for Jim. We are closing out NARC. We are winning the war on drugs. While we talk about science, talk about biology, we have journeyed through many topics.
Did anybody win the war on drugs?
No, it's fine. That $3 trillion was totally worth it. But yeah, that's why we closed with.
It was $3 trillion.
Some estimates say. I mean, when you talk about like incarceration for non.
Oh yeah. Well,
it's just like it is not cost effective. That combined with DEA and whatnot. Yeah, I used to have a PowerPoint slide where I was part of this Energy Frontiers Research Center where the budget was $10 million, which sounds like a lot, but it's supporting 60 people or something like that. But when you break it down versus the F-35 jet program, right, which is like 10,000 EFRCs, or versus the war on drugs, or versus whatever pet project people are doing.
ICE.
Yeah, ICE, exactly. Don't get me started. But we'll see what happens with those budgets.
Yeah, that's a
contentious right now. I mean, are you guys doing that math in terms of like recruiting students and admissions and stuff?
Yeah, so it's a lot of our students can be supported by TAs, but it was difficult to recruit after COVID, and many of our colleagues' universities stopped recruiting efforts.
Wow.
Because of Doge and cuts to federal funding.
Yeah.
And so our carefully laid plans don't always end up as well laid when the whole system changes at once.
Yeah, and for us, I mean, the can't have Chinese, Russian, North Korean, Venezuelan, Cuban. Like that devastated a non-trivial portion of our physical chemists.
Computational work, biologists, people interested in biotech. It's I'm sure there's easier ways to get spies into the United States than packaging them up as a graduate
school.
student.
Yeah. Why make them suffer?
That's right. Yeah, who's going to sign up for that one?
Yeah, exactly.
We're going to send you to America to steal trade secrets, but you have to be a graduate student. So, no.
It'll be 5 years, but it'll be fine.
That's right. Gulag. I'll take the Gulag. I hear the outcomes are better.
Oh, Texan, by the way, I'm liking the new disguise, dude. Facial hair fits you well. Well, thank you. It became a laziness thing at some point where I just got sick of shaving. But no, I appreciate it. My wife really likes it. I get annoyed at some point. I don't know if you have this issue, Jim, but if it gets too long, I'm like, it's gone. I can't deal with this anymore.
Oh, no. I'm so lazy.
Yeah.
Last time I shaved it, I think I had my oldest boy was like 2 or 3 years old. He had never seen me without facial hair, and he didn't know who I was. So I grew it back, and it changes lengths.
Yeah.
Doesn't have to get very long in Florida.
Yeah, but then you have to like maintain it, right? A certain length, and
Yeah, you just buy a set of dog clippers or something.
Yeah.
It's easy.
that's awesome. Yeah, we'll have to see. This is Evil Ken.
Oh my
If I've learned anything from Star Trek, it's Evil Jim and Evil Ken.
No, now it's turning in, like, I have more gray in my beard than I have gray in my. Well, I don't have as much hair
That
on
is
my head
intriguing.
anymore.
What are your thoughts on nature versus nurture on that front?
On grayness,
Yep.
I really haven't thought much about it because I lost my hair faster than it grayed, so it was a non-issue
Oh,
until the beard started getting gray.
mine's going reverse sequence. It's gonna go eventually, but it'll be white for a while.
It's not coming back.
Oh man! And if you're sick of this, I'm happy to intervene.
Yeah,
It is
please
getting
do.
Late. So this one, it's so dumb, and the reason it's dumb, I'm gonna show you guys this so you guys can learn about NARC. Is if you turn around and line up with him because of where the hitboxes are, you never get him.
Right.
So what you have to do is basically either shoot him like this and move up and down and hope one of them hits. But what I found out is if you line up right here, it's the perfect position on the screen where you line up your y-axis with his y-axis, and then you
And then
can
it
get him.
picks them all.
Yep. So it's just a very subtle. So
That's
now you have the
interesting.
card, you go
Yep.
through it. If you're interested, there's a half-hour tutorial on YouTube that I made telling people how to beat this game. So, civil forfeiture to the level, and we're going to close out NARC with a victory.
Let's get some gold.
Yeah, might as well. You earned it.
I don't know if I earned it, but it's my only opportunity in this lifetime.
Yeah. What I'm impressed with is those are $24,000 blocks of gold. They got to be heavy. All right, Jim, congratulations. 30 minutes and 31 seconds. I think you beat your previous time by 6 or 7 minutes. So we're going to call this a victory for everyone involved. Yeah, if you guys have suggestions who we should raid, I didn't even look who was on it. I didn't ask earlier. I probably should have. But yeah, if you have suggestions, put them in chat right now. But anyway, Jim, it's been 4 years. Return visit. I'm really glad you did. It's been a
Let's
lot
not
of fun.
make it 4 again.
You get to put your initials in there. Well, what ends up happening is it's 8 guests per semester. And so cycling through, we've had 80 different people on screen.
Okay.
There we go. Jim has made the scoreboard several times tonight. Between this, go down to end with that mutated finger.
Yeah, it's kind of a decrepit finger.
All right, there you
Not
are,
sure
Jim.
my finger's that color.
Well on top of the scoreboard, winning by 2 orders of magnitude against the previous scorekeepers. But Jim, thank you again for joining me after your sabbatical. We negotiated several times to get you on, but I'm glad we did. It's been a lot of fun.
It was fun. This is the most anxiety I face in my life.
But it's never bad as you think it is, right?
No, it's like getting a filling.
Yeah. Is it? While you're on.
No. Getting the filling, Novocaine.
Yeah, exactly.
You don't feel it anymore.
We anesthetize with ethanol here, but we do our best. But it was a lot of fun. We went through a bunch of games. We played Galaga. We played, what did we go through?
Centipede.
Galaga, played
Star Wars.
played Close the Night with NARC. It's been a good night. Thank you all for joining us. Oh, Cuddle Puppy looked it up. 24,000 in gold is 7.2 ounces. So those bars weigh less than half a pound.
Well, that's because gold is just crazy inflated right now.
Yeah, fair. But what was it in 1986 when this game came out?
Yeah,
That's the real
It was
question.
probably about 260, maybe $300.
Yeah. We got to do that. Oh, there we go. Science stream Doomfruit. I don't know who Doomfruit is. So we have someone doing art on stream. Should we do, what do you think? Art
for the
or
next
science? Yeah.
Yeah.
All right. But anyway, thank you all for joining us. Thank you. It's a pleasure. We're about to cross 950 followers on stream, and it's been a slow grind over 4 years, but I think you're our 109th stream, which is pretty incredible. You were one of the earliest ones that joined us, and I'm glad you were able to come back and join us. Any parting words for the audience, either biology or otherwise?
No, just play some games. It's actually relaxing. This is more relaxing than getting to filling.
That is the best endorsement we've had for Ask a Scientist Gaming. Tell your friends and colleagues they should come on. Tell Debbie she needs to. Like, we'll find a game. I promise you, we have something she wants to play.
I mean, chess.
Yeah, we can do chess. We should have done pinball. We didn't do that one.
There's got to be something out there.
Yeah. Next time I cross paths,
I'll harass her.
I'll see if she's interested. But anyway, thank you all for joining us. This is our last stream of the summer. Unfortunately, I don't have the fall stream solidified. I try to put those all together at once, but I'll put them up on the about page under schedule. You'll find a box there. But I do know who the next guest is on September 3rd. Rachel Yohei from the Department of Physics at Florida State University. She is a high energy particle physicist. In particular, she does the theory and analysis of experiments that occur at the Large Hadron Collider. She's also involved in like developing those experiments, what detectors they use and how they process that data. And so, yeah, particularly interested in Higgs boson and all these particles that are generated. So join us again September 3rd. That'll be 2 weeks from now, and then I think we're pretty consistent from there out to every 2 weeks for the fall semester. So yeah, thank you all for joining us. Your questions have been awesome. Yeah, I look forward to going back through that list because it's been all over the place. So she's trying to turn the Earth into a black hole. You will have to ask Rachel in 2 weeks. I don't know what she's going to say, but I'm going to assume no, but maybe she is. But anyway, stick around. We're going to raid Education Art Streamer. We're going to figure it out as we go. But yeah, thank you. The questions have been great. It's been a lot of fun. Jim, thank you again for joining.
No, thank you. It was a lot of fun.
All right. We'll see you guys in 2 weeks.